But Sylvia, I think you can join us upstage to represent the ACE project. So Dr. Sylvia Nkandiwiri is the project manager for the ACE Impact project of the World Bank. Please join us upstage. We also have our guest of honor, Professor Olusola Oyewole, who is the Secretary General of the Association of African Universities. Prof, please join us. We have Monsieur Jules Ahmad Amniabosu. Yeah, you're represented by Mr. Florent. Yeah, please join us. And then we have our keynote speaker for this morning, Dr. Patrick Duffy. Please join us. Yeah. Okay. All right. Dr. Christophe Cote, the country director for AFD, please join us. I hope we have enough chairs. Yeah. I think it's enough, right? Okay, so I think we're set. So this morning I'm going to be co-emceeing with my friend, Professor Nikes Ndam, who is our collaborator and one of the RD lead persons in Ghana and now in Benin. So Nikes would also be helping steer this morning's program. So I would like to acknowledge the presence of some colleagues. Professor Mark Yidana is the Dean of the School of Physical and Mathematical Sciences. Okay, our Pro-VC is here, Pro-Vice Chancellor for Research, Innovation and Development, who's here to represent the Vice Chancellor. Please come upstage. Yes. So in the University of Ghana, we have two Pro-Vice Chancellors, so that you don't get confused. So, yeah, I'll sit here. So, he's my colleague. He's the Pro-Vice Chancellor responsible for Research, Innovation and Development. And I'm Pro-Vice Chancellor responsible for Academic and Student Affairs. So he's here to represent the Vice Chancellor, who I said is out of town. This morning, we are joined by our colleagues from the Creek Africa Network, led by Dr. Jean Langhorn. Jean is over there, and they are here with a very strong team from the Francis Crick Institute. And they'll be joining us for the next few days. And at the end of the program, we'll have some discussions about collaborating between Francophone and Anglophone countries. So, ladies and gentlemen, it's my pleasure to introduce you to WANIDA. So WANIDA stands for the West African Network of Infectious Disease, ACEs. ACEs stands for African Centers of Excellence. So the African Centers of Excellence, as you know, is a program that was initiated by the World Bank as part of their efforts to build scientific capacity on the African continent. And the main goal of the project was to provide specialized training through the establishment of specialized centers of excellence. And WACCBIP, where we sit today, where I'm the founding director, was one of the first beneficiaries of the ACE project. Subsequently, there was a phase 2 and phase 3, and as part of the phase 3, which is called ACE Impact, there was a partnership with IRD and AFD to establish what we call ACE Partner, which is a partnership network aimed at fostering further partnerships between African institutions. And so as part of that program, we put in a proposal to establish WANIDA. And so WANIDA is part of ACE Partner, which is built on the ACE project. So you're going to be hearing a lot of ACE. ACE stands for African Centers of Excellence. So WANIDA had 7 founding partners. I would mention their names, and we welcome their teams that are here. Each of them are represented here by a team of scientists. So, of course, WACCBIP is the host. So, WACCBIP, where are the WACCBIP reps? Including myself, WACCBIP, please wave. Then we have ACEGID, African Center of Excellence for Genomics of Infectious Diseases in Nigeria. ACEGID, we have ACEGID here. ACEGID is over there. Welcome. We have African Center of Excellence for Mycotoxin and Food Safety. This is also from Nigeria. Then another one from Nigeria, African Center of Excellence for Neglected Tropical Disease and Forensic Biology. Yeah? Strong team. Then from Burkina Faso, we have ITEC MTV, which is based in Bobo Dioulasso. ITEC MTV. And then from Guinea, we have the African Center of Excellence for Prevention and Control of Disease Transmission, which is CPCMT. Our friends from Guinea, yeah? All right, great. So we have... Oh, did we miss Kanu? ACEFAB, we missed ACEFAB. ACEFAB is one of our most active centers, sorry. ACEFAB is from Bayero University, yeah? So African Center of Excellence for Population Health and Policy, ACEFAB. Yeah. Professor Maman, did I mention you? Okay. Okay, I didn't forget you. Good. All right. So we have representation from the Association of African Universities, World Bank, GTEC. Edmond is here on behalf of GTEC. Dr. Edmond. I don't want to try your name again. Yeah, but Edmond is from GTEC and is the project coordinator for the national level for the ACE project. So, have I left anybody out? So, everybody is very welcome to WACCBIP, very welcome to University of Ghana, and welcome to this conference. And we look forward to having a good time all together in the next 3 days. Nikes, please come and fill in the gaps if I left some.
Yeah, just to fill with 2 new centers that joined the WANIDA.
Yeah, go ahead.
Yeah, these are IRCB from Benin. I think they are there. And Sermes from Niger. Ronan is there.
Great. Good. So for WANIDA, even though we have the 7 founding members, because we want to build partnerships across the continent, we open it up for institutions or centers or partners to join. And so you need to just express interest to join. And the WANIDA partners would consider what value you bring and what value we can give you, and then we accept you into the network. So the last 2 that Nikes introduced are our new members who have joined us, in addition to the 7 founding members. All right, thank you. So we would go ahead and take some short remarks from our guest. So our landlady, the Vice Chancellor, is not in town, so she's being represented by Professor Felix Asante, who's our Pro-Vice Chancellor responsible for research, innovation, and development. So he would welcome all of us with some brief remarks. Thank you.
Good morning, colleagues. On behalf of the Vice Chancellor, who would have wished to be here, but she's currently out of the country. And on behalf of the management of the University of Ghana, I wish to welcome you all to this network symposium. I've been following as responsible for research. I know when this network was formed, because I know we had to issue sub-awards and contracts to member institutions, and I'm happy that WACCBIP is hosting this symposium. The topic, infectious disease in an era of global change, innovative, multidisciplinary, pan-African efforts towards building research capacity in Africa. This is a mouthful. I know it's only the scientists who can say this with ease. I'm a social scientist. That's why I found it very difficult. But I think the topic is very, very timely. And I must say the whole area of infectious disease is a priority area for University of Ghana. We are currently working on our new research strategy, and infectious disease and communicable diseases features very, very, very prominently. We encourage multi and transdisciplinary research, and I'm hoping, and I know Professor Wandari is encouraging that in this symposium, and also how you tackle research topics in this area. So once again, on behalf of the university, I wish to welcome you all. Please find time and visit our beautiful campus. And if you have any questions on why the university is this, Professor Wandari will answer it for you. So once again, welcome, and please, we are at your service, and we are happy that you are here, and enjoy your stay at the University of Ghana. Thank you.
Thank you very much, Felix. Please, Ekua online would quickly take her remarks if she's online. Is she there?
Can you hear me? Sylvia, I'm connected.
Okay, all right. So it's my pleasure to introduce Dr. Ekua Bento. Ekua Bento is a senior education specialist at the World Bank and the task team leader. These are terminologies from the World Bank. Task team leader basically means the person who's responsible for the World Bank ACE Impact project. So she's all of us our boss. And she's also from Ghana. So we're proud to have a Ghanaian holding such a significant position at the World Bank. So Ekua, please, your remarks. Thank you.
Can you hear me okay? Can you hear me? Okay, I believe you can hear me, so I'll go ahead. Thank you very much for the introduction, Prof. Arandari. I would like to just say a big thanks to also Prof. Oyewole, the Secretary General of the AAU, the AFD and IRD representatives, all protocols observed. It is a pleasure to be here to offer a few opening remarks to open this very important symposium. Thank you for the invitation. I apologize for not being able to be with you physically in Ghana due to prior engagements, but I'm glad I'm able to join you virtually, although it's very, very early here in DC. And being from Ghana, I know that I'm missing out on the warm weather and some good Ghanaian food. The topic you have chosen for this event is very important and timely, and I'll keep my remarks very brief. I am particularly excited to see where it's like innovative, multidisciplinary, Pan-African in there. And it is obvious that together we can be much stronger, we can do more faster than the individual parts. I therefore applaud the WANIDA network, which is one of the most dynamic networks in the set of networks that we have under the ACE Impact Project, for picking this topic and striving to achieve this within the network. These thematic networks were included as part of ACE Impact to ensure that as a collective and multidisciplinary network, you'll be able to find common work together within the ACE family, but also bring along other partners nationally, regionally, and globally. Today, the world is dealing with several challenges that know no boundaries and therefore require efforts from across the globe. Africa, which bears the brunt of many infectious and other types of diseases, can therefore not be left behind in these efforts and must be part of the solutions, and not only as a testing bed, but as part of the scientific engine that drives research behind these solutions. It is my hope that we are able to go beyond the lifetime of the ACE project in terms of the strong scientific collaboration that is being built within these networks. More and more, I have been receiving a lot of requests for partnerships on ACEs, and I frequently say that the ACE centers always make my work so much easier, because we no longer have to struggle to make the case that our ACEs are making strides in delivering on innovative and quality research. So thank you. I wish you the very best in your deliberations during this symposium. And hopefully I'll be able to join you again during the closing, but I have other engagement. I'll try my best, but I wish you the very best for the next couple of days as you come together and discuss your research and form various different partnerships. Thank you.
Thank you very much, Ekuya. I think I missed to introduce one of our friends who's joining us from Harvard, Dr. Jacques Kodbonu, who's also a Ghanaian and who has been engaging with WANIDA, trying to build partnerships. Yeah, and Gregory needs to also be introduced. Gregory is the project manager for a project coordinator for the ACE Partner Project, and Olivia, who's sitting next to him, is the project manager for WANIDA, who put all this together, who organized all this. So, Olivia, please take your bow. All right, so we'll move forward. We'll get some remarks from Florent. Your turn? Okay, thanks.
Okay, ladies and gentlemen, dear colleagues and friends, all protocol observed. On behalf of His Excellency Jules Armand Amiamboso, Ambassador of France to Ghana, it's my pleasure to welcome you in Accra on the impressive premises of WACCBIP, so the West African Center for Cell Biology of Infectious Pathogens of the University of Ghana. His Excellency asked me to convey his sincere apologies for not being able to make it this morning due to previous commitments. I know that he's involved notably in several events linked to the International Women's Day, which takes place today. So this gives me the opportunity to wish all women present in the audience a very good Women's Day. This also, thank you. Gordon and Nika, this also gives me the opportunity to congratulate the symposium organizers, since I've seen in the program that many presentations will be made by women. So this is clearly the way forward. So I'm very happy to be here with you this morning for numerous reasons, both professional and personal. Because of time constraints, of course, I'll mention only a few. Firstly, this symposium is fully in line with the priorities set by President Macron at several occasions, and most recently during the press conference he held on the 27th of February on partnerships with Africa. He mentioned indeed that education, professional training, employment, health are, among others, priority topics for collaborations between France and African countries. Another reason is obviously the very high quality of research and training performed through WANIDA since its inception, with, I've been told, 19 masters and PhD students already trained in members' laboratories, the participation of WANIDA faculty in 36 international symposia and workshops, and the publication of more than 15 scientific papers co-published by WANIDA members, and of course with several more in the pipeline. Yet another positive aspect of WANIDA is its regional dimension, provided by the participation, as has been told already, participation of African centers of excellence from Anglophone countries, 4 ACs from Nigeria, 1 from Ghana, who is hosting us this week, and from Francophone countries, 1 from Burkina Faso and 1 from Guinea. Another good news is that WANIDA has expanded in 2022 with the integration of other key partners in West Africa, the IRCB, Institut de Recherche Clinique du Bénin, and the CERMES, Centre de Recherche Médicale et Sanitaire from Niger. One last point I would like to underline from a personal perspective is the strong involvement of French institutions, first the AFD, the French Development Agency, through the funding provided to the ACE partner program. But my colleague Christophe Côté, who is here with us this morning, will certainly address this issue during his coming speech. The second French institution I would like to acknowledge is the IRD, the French National Research Institute for Sustainable Development, for efficiently coordinating this ACE partner program. It should be noted also that INRIA, the National Institute for Research in Digital Science and Technology, contributes both to ACE partner and to WANIDA, in the latter case through the provision of artificial intelligence tools in the framework of a PhD project. I would also like to mention the strong support provided by the IRD researchers who are posted in the countries which participate in WANIDA, and notably the IRD team in Benin, and also particularly the IRD researchers who are present here in Ghana. Indeed, they have been instrumental in developing excellent collaborations with Ghanaian universities and research institutes. So they are hosted since 2015 on the UG campus at the Noguchi Memorial Institute for Medical Research. And I would like to take this opportunity to thank Boss Prof. Nana Abba Amfo, Vice Chancellor of UG. Please convey the thanks of the French Embassy to her. And also Professor Dorothy Yeboamanu, Director of the Noguchi Institute, for their continuous support, which makes this collaboration between Ghanaian and French research team a success. Before concluding this speech, I'd like to mention a more personal point. Before joining the French Embassy in Ghana as attaché for scientific and higher education cooperation 1 year ago, I've had the privilege to work for 4 years as the representative of IRD in Benin. So you will easily understand that I am particularly happy to welcome the delegation from Benin this morning. So dear friends, it is really a great pleasure to meet you again. In conclusion, on behalf of His Excellency Jules Armand Amiam-Bossou, Ambassador of France to Ghana, I wish you all a very good stay in this country and a very successful symposium, and I thank you for your attention.
Thank you. Thank you very much, Florent. So we would like to take some remarks also from the country director for AFD, Dr. Christophe Cotter.
Thank you. Dear participants, dear colleagues, dear friends, it's both a pleasure and an honor to be here today for the opening of this 3-day symposium. And I would like first to thank the WACCBIP for hosting us today. When AFD is for Agence Française de Développement, French Development Agency, decided in 2019 to support the ACE partner project with a grant of 6 million euros, I cannot understate how much convinced we were that it was important to have the WANIDA network in place to encourage greater cooperation on infectious disease between West African countries and West African research centers. But I must admit that the achievements of the project have gone beyond our expectations. I think we can. With the COVID-19 crisis, Center of Excellence in Health participating in the ACE Impact Program and the WANIDA network have been at the forefront of the emergency response. As just one example, and I think it's a good place to mention that, at the very beginning of the Ghana outbreak of the COVID-19, WACCBIP helped develop very rapidly cheap diagnostic methods which allowed for detections and diagnostics of the COVID virus. Thank you for that. The ACE Partner Project is an institutional cooperation between the World Bank, the Association of African Universities, the French Development Agency, AFD, the IRD, and INRIA. It aims to promote the outreach and collaboration of thematic research networks between African Centers of Excellence, ACE, the key actors in quality education and research mobilized around national and regional development issues. WANIDA network aims at developing mechanisms for sharing resources between West African researchers and technical expertise. This high-level symposium showcases that wish, I think, which is to bring together renewed experts and practitioners to discuss research and its impact on crucial issues for Africa, such as surveillance, preparedness for emerging pandemics, disease prevention strategies, and vaccinology, neglected tropical disease, etc. Let me also take the opportunity of the event to say that AFD supported 2 other projects in partnership with the WACCBIP. In 2020, the IACOF project, coordinated by IRD, financed seroprevalence studies in the Greater Accra region to analyze the dynamics of SARS-CoV-2 infection. It highlighted the higher than anticipated level of prevalence of the virus in the population, in the Ghanaian population. 13.8% in November, 40% in July 2021. This project has been prolonged by the AfroScreen project to enhance genomic sequencing capacities of 13 African countries. Let me take this opportunity to salute the quality of this research partnership between internationally leading researchers playing a key role in supporting evidence-based decision-making. To conclude, thank you WACCBIP, ACE partner teams, and let me thank especially Olivia Koupaki, coordinator of the WANIDA network, for its hard work in setting up such a high-level event. We all look forward for participating to the plenary and keynote sessions, and we are confident that this event will bring further collaboration at a Pan-African scale. Thank you.
I would like to crave your indulgence to draw attention to a technical issue with the interpretation device. So we have two devices. One is Bosch. So if you are using the Bosch device, channel 1 is for English and channel 2 is for French. And if you are using the Brala device, channel 2 is for English and channel 3 is for French. So please take note. Thank you.
Okay, good. Thank you. Thank you very much, Christophe.
You should say that in French also. I can do that for you.
Okay, please help us. I was going to try, but it's okay. I'll let you.
Juste une considération technique. L'appareil de traduction simultanée, le canal 1 est pour l'anglais et le canal 2 est pour le français. Merci beaucoup.
Merci beaucoup. All right. So we're going to take remarks from our guest of honor shortly, but continuing with my introductions, I want to introduce David, who's currently the RD resident officer here at the University of Ghana. David. I also forgot to introduce my colleague, Dr. Peter Kwashi, who is a senior research fellow here and deputy director for research at WACCBIP. And we are joined by our head of department for chemistry, Dr. Mary Chama. Okay, so it's my pleasure now to introduce our guest of honor, the Secretary General of the Association of African Universities. The AAU is where the World Bank ACE project is hosted. And so he is also one of our big bosses. So my pleasure to welcome Professor Olusola Oyewole, Prof.
Good morning, ladies and gentlemen. I bring you greetings from the Association of African Universities, and permit me to also join others in congratulating our women on the celebration of the Women's Week at this particular time. Congratulations to all our women. I'm greatly honored to be part of this very important meeting, which happens to be the maiden symposium of the West African Network of Centers of Excellence on Infectious Diseases, WANIDA, seeking to bolster Africa's research capacity in tackling infectious diseases in the region. Ladies and gentlemen, we recognize the wind of global change that has ushered the world into an unprecedented era of technological, demographic, and climate transformations, as well as an increased risk of infectious disease outbreaks. We all will agree that the 21st century has witnessed a wave of severe infectious disease outbreaks, which has a devastating impact on lives and livelihoods around the globe. Africa has had its share of diseases peculiar to the region, including the Ebola virus, the Lassa fever, Zika virus, among others, which resulted in substantial morbidity and mortality in multiple African countries. This has necessitated concerted efforts to build capacities in combating infectious diseases in the region. The need to provide African solutions to African problems has become key to Africa's sustainable development and growth. I should mention that one of the lessons that COVID-19 had taught us in Africa is the need for African governments and African institutions to build their own capacities to meet the challenges of their time. Ladies and gentlemen, considerable efforts in scientific research and innovations have been made as contributions to global efforts to reduce the burden and threat of locally prevalent infectious diseases in Africa. The Africa Higher Education Centers of Excellence project is a supporting force in strengthening the capacity of African universities to address the developmental challenges in priority areas which include infectious diseases. I'm glad to note that very exciting outcomes have been recorded from the project supports in this regard, and this could be attributed to almost all of us present at this meeting today. I must commend the World Bank, the AFD, IRD, and of course the AU in the implementation of the ACE Partner Project, which has initiated 4 networks in demanding priority areas such as infectious diseases, mining and sustainable environment, sanitation and water, power and energy, and has also led to the development of other 8 networks funded by the World Bank ACE priority in the World Bank ACE priority areas. These networks have fostered inter-ACE collaborations in addressing developmental challenges unique to the African region. And I believe that this partnership will provide some sort of leverage in securing funding and building relevant partnership with non-ACE institutions and other networks with aligned visions, just as we've seen in WANIDA. I would like to emphasize that to enhance research excellence and strengthen research capabilities, the need for regional collaboration and adopting innovative multidisciplinary approaches cannot be underestimated. Ladies and gentlemen, apart from funding opportunities, networking platforms that promote regional collaborations among researchers remain very crucial. It is in this line that at AAU, just in December, we initiated what we call the African Research and Innovation Development Networks, AFRID. Our intentions in raising up AFRID is to see how we can bring African researchers working in the same area in the whole of the continent to work together. One of the other things we are doing in the AFRID network is that we are challenging universities to recognize their areas of strength and brand themselves in these areas of strength. We hope that with this, we'll be helping universities in Africa to use research to contribute to the development of their own region. It is my belief, ladies and gentlemen, that for Africa to move forward, for us to survive at such a time as this, let us work together. Collaboration is very important, and we need to network with one another. And I believe that these are the focus of WANIDA. Let me conclude by appreciating in absentia the Vice Chancellor of the University of Ghana, Professor Amfo, and the WACCBIP Director, Professor Wandare, for hosting such an important meeting. Further thanks to all stakeholders and centers gathered here today. I'm hopeful that the discussions to be held this week will be the genesis of greater works in the infectious disease landscape. Before I leave, I'll be glad to see many of you visiting us at the AAU. You are welcome, and to let you know that we are available to work with many one of us. I wish all of us a very fruitful deliberations. Thank you.
Thank you very much, Prof. Prof has summed it up well. We need to work together. And that's what WANIDA is about. So we would conclude this session of remarks, and then after that we'll take the keynote address or the keynote lecture from our colleague from the NIH, and then after that there will be a family photo outside and coffee break. So in that order, so we run a little bit behind time, but we'll catch up. At this point, I think that for the sake of the presentation, we would probably have to sit this way to watch Patrick's presentation. Whilst we move, I will call on Nikes to introduce our distinguished keynote speaker. Those of us who are students of malaria research, we've watched and admired Dr. Duffy for many, many years. And so at WACCBIP, we're really honored that he made the time to join us here. And we look forward to having good discussions about collaborations with him. He's also currently hosting one of our young research fellows, Dr. Henrietta Mensah Brown. Hetty is doing her NIH active postdoc with Patrick, and I'm hearing that they are doing great things together. So hopefully it forms a foundation for Patrick to return to Ghana and work here. So thank you. Nikes will do the formal introduction. Thank you very much.
The representative of the institutions, founders from the embassy, French embassy, and AFD, dear colleagues, dear friends, all protocol observed. After this first part of the official introduction of the WANIDA Symposium, I now have the privilege to introduce the first keynote speakers. So, Professor Patrick Duffy. And before that, I will have to acknowledge the presence here of Professor Koita from Mali, Professor Richard Njouom from Pasteur Cameroon, Professor Sami Amidou from Venda University, South Africa, and Dr. Claude Evray from Merck, and Jane from Crick Institute Network. So I have the privilege to introduce the first keynote speaker, Patrick Duffy. I knew Patrick through his work on placenta malaria, his work on Michael Fried, who were the first to elucidate the molecular mechanism underlying the pathogenesis of placenta malaria. So Pat and Michael were the first to identify the receptors, chondroitin sulfate A, that mediates the placenta tropism of infected red blood cells. And this discovery impacted the work of many researchers, including mine, and they put us on the track of the vaccine research aimed at protecting the sensitive organs of the host. So this is at the base of emerging vaccines on placenta malaria, and Patrick is the chief of the Laboratory of Malaria Immunology and Vaccinology at NIH. And he is also, as Gordon was saying, he is a pioneer. He has pioneered a path in partnership with Mali colleagues to test most of promising malaria vaccine in pregnant women that they are developing. And I will therefore leave the floor to Pat Duffy to take us through the topic of malaria pathogenesis and vaccine discovery. Patrick.
Okay, well, I want to thank Nikes and Gordon for the invitation to join. It's really impressive to see what WANIDA is And that was one of the encouragements that Nikes gave me, was to come and see this amazing network of scientific groups across West Africa. We've been heavily involved in training ourselves of US scientists, but also many African scientists. So I'm really inspired what everybody's doing, including the funders who provide the resources to make all of this possible. So Nikes and Gordon asked me to talk about how pathogenesis research is related to translating discovery into products for people. So that's what I'm going to talk about today. Now, how do I control this? This one? Yes. So I'll quickly talk about malaria epidemiology and disease burden and how that informs studies of pathogenesis, including host-parasite interactions. I'm going to focus on what Nikes talked about, which is pregnancy malaria, and how research over the past 30 years puts us on the cusp, we believe, of a vaccine that can protect pregnant women, their unborn children, and their newborns from disease and death due to pregnancy malaria. And then finally, I'm going to touch on severe malaria epidemiology and how that informs pathogenesis and how we should be thinking about a vaccine for children also. So let me start with malaria epidemiology and disease burden. Malaria, for those of you who are not working on it, has some distinctive epidemiological features which really tell us something about what the underlying disease is. This works. So in general, during the earliest months of life, you're protected, but then there's a pretty rapid increase in the incidence and prevalence of infection. And this really lasts for many years until about adolescence, when there's gradually an improving control of parasitemia that you measure by taking blood smears from people. And then you get to an adult level of protection where you continue to be infected, but you don't get sick, and it's not infected as frequently. There's a very distinctive feature, which is it's severe malaria, which actually occurs during a relatively brief window of life. So if your child lives to be 5 years old in many of these communities, they're not going to die from malaria. And that is responsible for 500,000 or more deaths each year of primarily African children. That looks like a vaccine because it is controlled relatively quickly, but we don't understand why it's controlled so quickly, so it's not yet a vaccine. Another distinctive feature is what happens in women when they become pregnant, because like other adults, they control malaria. But when they have their first pregnancy, there's suddenly a renewed susceptibility to infection. And then that susceptibility appears to decrease over successive pregnancies. And when myself and my colleague, Mikhail Fried, that Nikes mentioned, entered this field back in the early 90s, it was thought that pregnant women became susceptible because they were immunosuppressed or immunomodulated during pregnancy because the fetus is a foreign entity. But that wouldn't explain why control would improve over successive pregnancies. So it puzzled us and it inspired us to work with colleagues to study this problem. I guess I have to do that. Here we go. So first of all, to recognize that this is a problem that is not captured in WHO numbers. So when WHO tells you that there's 600 and something thousand malaria deaths every year, they're not including the deaths that are related to many of the deaths that are related to pregnancy malaria, because it's difficult to see those. In high transmission areas, women that become pregnant have systemic immunity that prevents them from having many symptoms. They nevertheless have an insidious development of severe malarial anemia that it can contribute to deaths around the time of delivery due to hemorrhage. And then we also provided evidence that gestational hypertension. Thank you. Is another aspect that I'm not going to be talking about. For the developing fetus and the newborn, the most measurable and first identified problem was low birth weight in the offspring and preterm delivery that predisposed them to mortality during infancy. And in studies that have looked at chemoprophylaxis studies, about a third of perinatal mortality is related to malaria in these communities. Let's see my picture appear down there. In low transmission areas, women don't have systemic immunity. So actually what you see in those populations is something that looks like what happens in children. They develop severe syndromes and they're at higher risk for cerebral malaria, respiratory distress, and acute severe malarial anemia than other members of the population, non-pregnant women. And there's been recent evidence in these populations that they also suffer from gestational hypertension or preeclampsia related to Plasmodium falciparum malaria. But for the offspring, you see similar sequelae, which is low birth weight, abortion and preterm delivery, and neonatal and infant mortality that is clear to see because the women are having severe episodes themselves. So you can relate those poor outcomes for the offspring to the actual malaria case that the woman's having. The estimates are about 50 million women in areas of high transmission area are exposed every year and 75 million in low transmission areas, but the greatest burden is in these high transmission areas just because of the frequency of malaria in those populations. And in Africa, the recent estimates are there are up to 50,000 women who die, and there are 200,000 stillbirths. And I mentioned to you that also low birth weight and preterm delivery are predisposing the offspring to neonatal and infant mortality. That's hard to measure because the women didn't feel very sick. So it's a heavy burden of disease, and it's really not captured by those numbers from the WHO. So we actually have been, this is my colleague Mikhail Fried, who I've been working on this problem for 30 years. We started this work as part of the Kenya Medical Research Institute, and now we have a strong partnership with our colleagues in Mali, and specifically Professor Alassane Diko is leading these studies. But one of the things we've done is to try to improve the ways that we can capture information about women's malaria episodes and tie them more closely to the poor outcomes for women and offspring. And we do this by intensively following women for malaria episodes. And what's important to recognize here is that Mali is a place where antimalarial drugs still work, sulfadoxine, pyrimethamine, and bed nets work, and they're pretty well applied. Despite that, we can see that most of the women in our populations that we follow have malaria during pregnancy, despite the application of the available tools that are recommended. And because of its frequency and the intensity of our studies, we have been able to show that malaria infection is common and associated with perinatal and perinatal mortality and preterm delivery, despite the fact that these women were getting the measures that are recommended for them. And what we also showed is that this relationship between malaria and poor outcomes is related to systemic inflammatory responses during malaria episodes that appear to be associated with these poor pregnancy outcomes. And this is the kind of evidence. This is actually evidence that Mikhail and I developed when we were working as part of the Kenya Medical Research Institute, showing that in children that are born low birth weight, as an example, TNF-alpha levels are significantly higher in the placentas of those women than children that are born normal birth weight in an area where women are being exposed. So there's a relationship between infection, inflammation, and the poor outcomes, both for the women as well as their offspring that I was telling you about. So just to describe pregnancy malaria to those in the audience that are not familiar, it has this distinct parity-specific epidemiology of risk during first pregnancy that decreases over sequential gestations. Pregnancy malaria syndrome and presentation depends on the state of the mother's maternal immunity, the amount of anti-malaria immunity that she has acquired over her life. But in both low transmission areas and high transmission areas, even if you have systemic immunity, pregnancy malaria is causing maternal, fetal, and newborn mortality, and that is related to placental inflammation. So now let me talk to you about, with that framework, how studies of pathogenesis have informed the effort to develop interventions. So this is what placenta malaria looks like. It's called placenta malaria because the parasites accumulate in the intervillous spaces. This has been recognized since about 1900, not long after Laveran discovered the malaria parasite in Algeria. So what you can see here are mature forms of parasites that are accumulating in these intervillous spaces. And actually, the work that Mikhail and I did speculated that this must be a specific binding event and that this was a distinct parasite phenotype, and that's essentially what we found out. We work with Dr. Alex Masori at the Nyanza Regional Hospital in Kisumu, and enrolled women who were just coming in for their deliveries and who would donate their placentas so we could study the parasites that were infecting the placentas. And what we found really quickly, within a few weeks, was that the parasites had this property of binding to a section of a placenta that's not infected. So we would make cryosections of uninfected placentas, allow parasites to settle onto the tissue and wash away what didn't bind. And what you can see is what we found was that parasites would bind right along the surface of the villus that's in the placenta. That's the fetal tissue in the placenta, but they're binding at where there would be an interface with the maternal vascular space. So this would be where the maternal blood had access. And this was later studies when we realized what it was binding to. Nikes already mentioned to you chondroitin sulfate A. So if we treat that placental section with chondroitinase to remove this glycan called chondroitin sulfate A, the parasitized red cells no longer bind there. And so pregnancy parasites bind to chondroitin sulfate A, and that distinguishes them from parasites that you collect from other people. So this is just an example of 2 parasites we had from placentas, 2 that we had from males. CD36 is a very common receptor that supports the binding of parasites. We have many more data, other groups have data, that if you collect parasites from placentas, they uniformly bind to this chondroitin sulfate A, and it's a very uncommon phenotype in other populations. So what happens is during pregnancy, you get a new organ, it has a different receptor, and it selects a distinct parasite phenotype to overgrow in the placenta. And so CSA binding parasites sort of became a shorthand for the parasites that would infect women. And I don't want to go through this data in great detail, but essentially what we found out, what this shows is the level of parasite binding that are Kenyan parasites that we collected from pregnant Kenyan women, and looked to see whether sera from first-time mothers, second-time mothers, or third-time mothers would reduce that binding to this placenta receptor chondroitin sulfate A. And what you can see is indeed sera that we collected from women in Kenya, first-time mothers did not inhibit the binding, whereas the multigravid women, their sera would inhibit the binding. We also found the same phenomenon with parasites from Malawi and parasites from Thailand. Women, study participants who had never been pregnant had no activity to reduce that binding. And so we found the same phenomenon when we worked with our colleagues in Thailand who provided us with parasites from Burmese women who were living in Thailand. This is Francois Nostin and his group. We found the same phenomenon with the same sera. And so what's important to recognize here is it appears that there's a very conserved process going on immunologically around the world where women are acquiring antibodies that are not only effective against the parasites that they might get in Kenya, they're also effective against the parasites that might occur in Burma. And this to me sounds like a vaccine for pregnant women, because if we can induce that, what we can do is to reduce the incidence of infection. And what I show you here is the data in Kenya showing that, first of all, no primogravid women had these antibodies there, and all multigravid women had these antibodies. Only in secundigravid women that there was heterogeneity in whether they had these antibodies or not. And women who were infected during their second pregnancies were less able to control parasite binding, linking those two things. And so this is now the immunologic feature that we would want to use to reproduce with the vaccine. And so what I've told you in this section is that Plasmodium falciparum parasites sequester in the placenta, and those parasites specifically bind to a distinct receptor there, chondroitin sulfate A. Women acquire antibodies naturally that block parasites from binding to that receptor. And the pattern of those antibodies explains why women are most at risk during first pregnancies and they develop resistance over sequential pregnancies. So now let me talk about vaccine discovery for pregnancy malaria, and a very important discovery that our colleagues in Copenhagen made. This is Ali Salanti and Tortiander. And what they were doing was studying a very important variant antigen family, variant surface antigen family of Plasmodium falciparum parasites in general, called PFE and P1. There's about 60 members of that parasite family. They took a well-characterized parasite in the lab, selected it to bind to this placental receptor. All Plasmodium falciparum parasites have the potential to bind to that placental receptor. It's not a genetically distinct parasite. It just puts up a different member of this family on its surface. That's it. and that is what's called VAR2-CSA. So if you select parasites to bind to CSA, it's that single member of this highly variant family that always comes up, and it's unusual because it's the only member of that family that you can recognize as a conserved entity, conserved in the sense of recognizable in that family. So something we still don't understand the genetic basis, why it would have that and why it would bind to CSA. But nevertheless, that is the gene of interest, and that quickly became the leading target for a vaccine that Nikase and we have been working on since then. I want to briefly tell you about a new model that we've developed for this, because one of the problems with pregnancy malaria is we haven't had a good model to reproduce that specific phenomenon. So this is an Aeotis monkey, and what we have the privilege to do at NIH is to set up a breeding colonies of this in our research facilities, and so that we can get pregnant Aeotis and examine whether we can reproduce the features of human placental malaria. And so this is the basic design of the study that we started with about 10 years ago. We would pair a male and female together. At some point, the female would become pregnant. The gestation is 132 days, and when we started these studies, we were given this parasite, CS2, that is a reliable CSA binding parasite. And what we found briefly, let's see, is that I'm having some problems with that. Can you advance that? Can you go back? Yeah, you can just put that data there. So what we found briefly is that the parasites that we infected those animals with, you can see the binding. These are images of the binding assay, binding to a control entity, BSA, binding to the CD36 that I mentioned, or the binding of parasites that we collected from the monkeys, the pregnant monkeys we had infected. We do get CSA binding parasites out of these monkeys. The figure that you show at the bottom shows the placental parasites that we collect from the placentas of these infected monkeys always bind 100% to the CSA. Parasites we collect from their peripheral blood. Oh, okay. I've been asked to speak a little slower. But the peripheral blood, what you can see is sometimes you have CSA binding parasites there, and sometimes you have CD36 binding parasites there. So it's interesting. The peripheral parasites are different, not always coming from the placenta. And I just show, using antibodies, that the parasites that are in the monkey are expressing this VAR2-CSA on their surface. But this becomes a model now that we can test our vaccines with, because these are monkeys that we can reproduce the placental malaria experience in. This is the VAR2-CSA protein. It's a very difficult protein. It's very large and cysteine rich. Nobody realistically thinks we can make this as a full protein vaccine in terms of a recombinant protein. There have been entities that have been brought into the clinic that use a fragment of this. Our colleagues in Paris and in Copenhagen have brought these two vaccines called Primvac and Pamvac. We worked with them to test their vaccines in our monkeys. They were doing studies in humans and have completed those studies. And what's important to know, I won't go through all the details of this study, we would immunize, we would test the vaccine response, and then we would give the monkeys placental malaria, which is something, of course, you can't do in humans predictably. And so we could see whether there was a boosting response. But I will just sort of cut to the chase to say that the results that we got with the vaccine were very similar to the results that they got in humans, which is encouraging for the model, but not encouraging for that first generation of vaccines. What I'm showing here is sera that were raised from the two vaccines of interest, Pamvac and Primvac, blue and red. And then we had a candidate ourselves that was, I won't go into the details of that, and then a control malaria antigen. And what essentially the vaccine would induce antibodies that only recognized parasites carrying the form of VAR2-CSA in that parasite. But if you went to another parasite, there was no reactivity to those parasites. So it was very narrow. Did I do that? It was a very narrow level of activity, and when you looked at the functional activity, you can see the same thing, that this functional activity of blocking the parasites from binding in the placenta was limited to the parasites that carried that very specific form of VAR2-CSA. So this is not a vaccine that would widely protect pregnant women. And just to quickly mention that when we gave the animals placental malaria, when they had a placental malaria experience, it didn't do anything to broaden their vaccine response. It didn't provide them with broadly neutralizing antibodies. So one of the advances that we've had recently in our lab is our ability to make full-length recombinant forms of this that appear to be structurally correct. This just shows 7 different variants of VAR2-CSA on this gel. On the left side, it's non-reduced protein. On the right side, it's reduced protein. But now we have the material of VAR2-CSA to really work with and understand how immunity is working and how the protein is binding to chondroitin sulfate A. The figure on the right just shows the approach that we took to improve the expression of this protein using mammalian cells, which actually raises hope that we could make an mRNA vaccine out of VAR2-CSA. We've been able to use these for structural studies, and what I show here is structure that we obtained with our colleague Neeraj Tolya, who works in the office right next to mine. This is now the protein that's responsible for placental malaria, and what you can appreciate right here is chondroitin sulfate A actually threads its way through a pore, a channel that's created in the protein that involves actually a confluence of multiple of the domains of this very large cysteine-rich protein. And this is a schematic that shows that interface. Really, this is chondroitin sulfate A. This is a major channel that threads through, and then there appears to be a minor channel. So this large, complex protein folds around itself, creates a channel, and chondroitin sulfate A is a long chain that threads through that and allows the parasite to bind in the placenta. Some studies that one of our, I guess, mid-career scientists at this point, but Justin Derichmo, who's from Benin and worked with Nikes before he came to NIH, he used these to study how what naturally acquired protective immunity is. What I diagram here is Justin's efforts to take those multiple different variants of R2-CSA, purify antibodies on that protein, and also deplete them from the sera. And then you can use that purified antibody or depleted sera for studies that characterize the nature of those antibodies. And most importantly, this binding inhibition assay is how we test whether antibodies can block the parasites from binding to the placenta. And the important information, probably there's a whole set of data that he got, but the most important piece of information he got just by taking a single variant of VAR2-CSA, purifying antibody, and testing its ability to inhibit a wide array of placental parasites from binding to the receptor. And what you see here, it's depicted the opposite of what I previously showed you. The higher you go, the greater the activity of the sera. And what he showed is that with a single allele of VAR2-CSA, you purify antibodies that are reactive with parasites from around the world that infect pregnant women. So even though the vaccines that we've had go into the clinic so far, we're not able to induce this immunity, the full-length protein has epitopes of fully conserved antibodies. So this is very encouraging for a vaccine. So to summarize what I just told you, placental parasites, which are CSA-binding parasites, have VAR2-CSA on their surface. VAR2-CSA antibodies that block them from binding increase with parity, and that would explain why women become resistant over successive pregnancies. We have found that VAR2-CSA fragments that have been tested thus far don't induce, they induce variant-specific serum activity, but what we're looking for is broadly neutralizing antibodies. But a single full-length VAR2-CSA can purify those broadly neutralizing antibodies from the sera of multigravid women who have been exposed to malaria. And then finally, let me just touch on this, severe malaria epidemiology and what we understand about that so far, because that's another key target for malaria vaccines. This is a study that Brunner Gonçalves, a young scientist from Brazil, completed while he was in our lab, working with our colleagues in Tanzania, where we were doing studies where we enrolled children at the time of delivery. Women would come and deliver, and then we followed the children for the first 5 years of their lives. And the important findings really that came out of those studies, and it's just this one slide, is that parasite density does not explain severe malaria in children. Children don't have severe malaria because they have a high parasite density. It also showed that control of parasite density does not explain immunity to severe malaria. If you look at malaria episodes before a severe malaria episode and after, they have the same parasite densities before and after. And finally, the risk of severe malaria is constant over multiple infections in children. In other words, a children that has had 0 previous malaria experiences in an African community is at the same risk of severe malaria as a child that has had 5 or 10 episodes. You do acquire immunity over a span of years, but it's not from a mild malaria episode necessarily. So just to say that the epidemiology of severe malaria can also inform our pathogenesis studies and how we look to develop vaccines against them. This is our group at NIH, a lot of amazing scientists, and there's Michal, my colleague. And these are the leaders of the teams that are our vaccine development unit. And these are our colleagues in Mali, where we have, NIH has had a 30-year relationship with them, and it's just an amazing partnership that is truly collaborative and makes vaccine discovery and pathogenesis research move very quickly. This program, when I got there for vaccine development, started with Professor Ogobara Dombo as the lead of the vaccine trial, and he very sadly passed away a few years ago. And has been replaced in terms of the vaccine development with Dr. Asaka Sagara, one of the professors there. So I thank you for your attention. I hope that I conveyed that basic research turns into products that can help people.
Few questions before we go to the photo. Yeah, there is a microphone in the room wall. So, Women's Day, we will start with Jane. Sorry.
Thank you, Professor, for your nice talk. Very informative for me. I'm sorry. As you remember, I'm director from Burkina Faso, RSS, Institut de Recherche en Sciences de la Santé. I would like to know about altruistic vaccine. Here your talk is about placenta vaccine, but what thing I would like to know is about altruistic vaccine, transmission blocking vaccine. 10 years ago, we evaluated the efficacy of this vaccine in our lab from using the antibody from your laboratory via Himenu. So I would like to know the issue of this vaccine.
I didn't quite catch the question.
So my question is about the issue of the altruistic vaccine, transmission blocking vaccine.
Transmission blocking vaccine?
Yeah.
Oh, transmission blocking vaccine. Well, that's,
Yeah.
So there's been a lot of progress with, so transmission blocking vaccines work by targeting parasite proteins that are expressed while the parasite's in the mosquito.
Yeah.
And so what we've learned is you can vaccinate people with those proteins. They're on the surface of gametes and zygotes that only are accessible, in the mosquito. And we vaccinate people, they develop antibodies, and when the mosquito takes a blood meal, they take up those antibodies, and the antibodies attack the parasite in the mosquito.
Yeah.
And so the answer to your question is those vaccines have completed phase 2 testing,
Okay.
and they show a very high level of activity
Okay.
to prevent mosquitoes from getting infected. So the
Okay.
way we test the vaccine, this is ourselves and our colleagues in Mali, we feed mosquitoes on the vaccine trial participants on a regular basis. And so we found that it reduces infection events in mosquitoes by over 70%. Pretty consistently. So those vaccines are moving into phase 3 development. Is that what you're asking me?
Yeah.
Yeah. So those are now advancing in vaccine development.
Okay.
Happy to talk about with you some more. It's a very exciting area of vaccine
Okay, I'm
development.
interested to discuss
Yeah,
with you.
I think
Okay.
those vaccines can be combined with the R21 vaccine that was tested in Burkina Faso. Or
Yeah,
it could be
yeah.
combined with RTSS.
Yeah,
but
yeah.
they have to be given to everybody. So for those vaccines to have their effect, you have to vaccinate everybody in the community. It's community, it's herd immunity, but that's the way that they will work.
Okay, you can talk. After.
Yeah, we'll talk about
Okay,
that.
thank you.
Thank you. I think there was a question in the back.
All right.
Jean, sorry.
This is going to sound stupid. I should know this. These women have antibodies presumably to other VARs. So are they immune? So if you take away the VAR2 CSA and you stop the placental malaria, is there a chance that they can get seriously ill from a non-placental malaria, or are they all immune to the subsequent VARs that would be expressed?
So actually nature has provided an experiment in a way, which is the delivery of the placenta. And what's been described is after the placenta gets delivered, a woman's parasitemia usually resolves after that is cleared. That doesn't mean that they're immune, you know, they'll get reinfected still after pregnancy, but that the specific infection that is filled with CSA binding parasites, that will resolve as well. There's a period of susceptibility to the postpartum that's been described that's not really well understood. It could be because vascular spaces are modified during pregnancy, so maybe the chondroitin sulfate might appear in other vascular spaces, or it could be because of the blood loss during pregnancy, you know, leads to a rebound kind of reticulocytosis, and both Plasmodium falciparum and Vivex prefer younger red cells to invade. But those women would still have all their immunity to those other PFE and P1s. And as I mentioned, if you take peripheral blood from pregnant women, lots of times there's CSA binding parasites, but lots of times there's also CD36 binding parasites, and those are obviously sequestering in other vascular beds, not the placenta. So you'll see different kinds of binding parasite phenotypes in the peripheral blood of pregnant women. So they're still getting exposed. There's somebody behind.
Yeah, thank you for the presentation. I'm asking, what is the benefit of this vaccine to the mother? And then can it be adapted in the general population? Since this is a very serious problem in Africa. Thank you.
You're going to have to hold on. The acoustics here are not good. Did somebody understand the question? The acoustics are not good here. What was I'll have to ask the question again.
Okay, what is the benefit of this vaccine to the mother? Can it be protective to the mother, and then can it be adopted in the general population for other people, considering the seriousness of this disease in Africa? Can it be adapted for the general population?
I think I understand your question. This vaccine should be delivered to women before they get pregnant. That would be ideal. They should get this vaccine around the time they get human papillomavirus vaccine to protect them from, you know, other infectious agents. So I think that there should be a menu of health options for women who are entering the reproductive years. That's ideal. Part of the reason it's ideal is because women that are pregnant are getting infected at the beginning of pregnancy, and many of them don't show up until maybe the second trimester for their first antenatal visit. So we would want to protect them from malaria from the moment of conception would be ideal. So I don't know if that's the question exactly that you're asking. Yes, we want to be able to give it to pregnant women if they haven't gotten the vaccine, but the ideal thing would be to vaccinate women before they have their first pregnancy. That would have the greatest benefit for them. Thank you.
Thank you, Pat, for this very interesting, very interesting keynote. And we'll now move to get the family photo outside. And I also I have to acknowledge the presence here of Dr. Florence Pradel from Gabriel Network. So please, let's go and meet the other officials before they leave outside. Hey, there is also...
Hello, everyone. Can you please leave your headsets behind? Your translation devices, kindly leave them on the seat, and then when you come back, you take it. Thank you.
Ok. Juste pour nous indiquer, on peut laisser sur place, voilà, les écouteurs de traduction simultanée. Il n'y a pas de souci. Nous pouvons les laisser sur place. Merci à toi.
Can we all move to the front of the main biochemistry building in a timely manner so that we can take the family portrait quickly?
Hello, everyone. Can we please be seated?
All right.
You don't need to be so close.
All right. I think let's try to get settled. We're running a few minutes behind schedule, so we have quite a packed schedule, which is a good thing. But that also means that we need to be quite tight on time. So I believe Prof. Awandari introduced me earlier. It's my pleasure to introduce my colleague, Dr. Gloria Megache, who is a faculty member here at WACCBIP and at the medical school, and she'll be chairing this session. And one second. This session will be Emerging Health Challenges, Building Sustainable Systems Within the One Health Framework. And I'll hand it over to Gloria. Before I do, I pray that all the speakers keep to time and the chairs will make sure that that's what happens. Thank you very much.
Thank you, Dr. Kwashi. Right, so welcome to this session. Our first speaker for the plenary one is in the person of Dr. Issa Aliyu from ASPAP, Nigeria, who is going to talk on tackling SARS-CoV-2 infection in Nigeria to guide public health interventions. So Dr. Aliyu, your audience, and you have 15 minutes, please. 15 minutes presentation and 5 minutes questions and answers. Thank you.
Okay. So our protocol will be observed. We'll be presenting tracking SARS-CoV-2 infection in Nigeria to guide public health intervention. Okay, by introduction, in Nigeria, the earliest confirmed cases of COVID-19 happened on 27th February 2020 from an Italian student arriving from Milan at Murtala Muhammad International Airport in Lagos. And the active case finding of this index resulted in identifying more than 200 suspected individuals who had contact with him, and only one was happened to be positive out of those 200 suspected contacts. And though thereafter some related cases were reported, but today we have more than 267,000 reported COVID-19 cases in Nigeria. This figure is relatively very low compared to most countries, even among African countries. Okay. The relatively low number of cases in Nigeria might be probably due to the limited testing capacity across all African countries, and the fact that cases reported using qPCR tests might probably result in underestimation of the actual disease burden in all countries. Since in this type of test, it's only symptomatic subjects and or subjects who had history of contact with positive subjects are tested. So this might leave asymptomatic individuals and those who have mild disease unaccounted for. Okay. But seroepidemiological studies can give a true estimate of COVID-19 infections by checking in the blood of individuals the SARS-CoV-2 specific antibodies. These antibodies can be acquired during infections or by vaccination. So epidemiology can also provide critical information on the extent of COVID-19 transmission in the population, identify risk factors of infection, monitor vaccine efficacy, distributions, and guide vaccine strategies and other public health interventions. For this study, we aimed at tracking the transmission dynamics of SARS-CoV-2 in Nigeria to better inform public health intervention, which is aimed at reducing COVID-19 associated clinical illness and mortalities. By conducting a large-scale population-based COVID-19 serological screening to detect previous or current SARS-CoV-2 infection using rapid diagnostic kits. Okay. Okay, study protocol. After obtaining the ethical approval from Federal Ministry of Health and State Ministry of Health, and having collected permission from the head of each institution who conduct the research is conducted in 4 states in Nigeria, the one in the north, 3 others from the south. But the data presented here is from the Kano state, which is the state conducted in northern part of Nigeria. And the site of studies includes the communities, shopping malls, bus stations, markets, educational institutions, and religious institutions. Okay. This is the study protocol. Okay. For the serological studies, having obtained the written informed consent from all the subjects, we use a structured questionnaire to collect socio-demographic data of the individuals, and then the laboratory analysis was conducted in the field. So for the serological analysis, we collected blood from the patient at the field, and then they were tested using rapid lateral flow techniques. And then individuals who happens to be IgG negative and IgM negative will be counseled, and the result will be judged. This type of individuals, the individuals who had never had any exposure or encounter with disease or the virus, so they are not immune, they are not exposed, they are not vaccinated. So we counsel them, advise them on taking either vaccines. And then those individuals who are IgG positive and IgM negative, we also counsel them and give them the results. These are individuals who are supposedly exposed to the virus, either by infections or by vaccinations. We counsel them and give them the results. But by individuals who are IgG positive and IgM positive, these are individuals who are suspected to have a recent exposure to the virus, probably in the last 5 days. They have encountered and they develop immunity against the virus. And those who are IgG negative and IgM positive, these are individuals who are recently exposed to the virus, and they might be having active infection at the moment, and they are infectious, they can infect others. So these individuals qualified for rapid antigen testing. We collected their nasopharyngeal swab to do rapid COVID-19 testing at the spot. So individuals who happens to be COVID-19 antigen negative, we also counsel them and give them the results. In the counseling, we ask them to we advise them to take PCR testing. And individuals who happens to be antigen positive for COVID-19, rapid testing, where they are qualified for PCR testing. And then PCR negative will also be counseled and the result given to them. And the PCR positive, there are samples who qualifies for sequencing. Those who have CT below 30 will be qualified for sequencing. Here. This is the result interpretation I mentioned. So result and discussion. Okay. The characteristic of our patients, From shopping malls, 16.6% of our subjects were from shopping malls. Sorry, it's a mistake. This is 24% of them from the market. The list comes from the religious setting where we collected only 1.5% of the subjects. The age distribution among our subjects, individuals who age between 20 years to 39, they were 63.9% of total subjects, but at least those who are above 60 years. The gender-wise, our subjects are males were 56.7%, while females are 43% of the total subject we tested. For the highest level of educations, individuals who have offered secondary school have the highest number of participants in the study, while those who postgraduate study are the least in the study subjects. For the income status of the subjects, those individuals at the low incomes group have the highest frequency, which is 5.8%, and the least were those of higher income class, which is only 5.2%. The employment status of our subjects, individuals who are unskilled, has the highest frequency with 43.6%, and the retirees had the lowest frequency for our among the subjects. Okay, so the site-specific seroprevalence, our study shows that the individuals at the educational institutions has the highest higher incidence of the seropositivity. This is followed by individuals from the shopping malls, with the least coming from religious institutions. And multiple logistic regressions analysis of the risk factors of positivity with the sampling site shows that it is more likely to be seropositive for COVID-19 if you are sampled at educational institutions, and then followed by shopping malls than other sites. This probably is due to the fact that these are the sites that's enclosed sites, so the chance of getting infections in this site is higher than others. Like for the communities, it's a bit higher, but it's not as high as the more enclosed site. The bus market stations also the positive having positive seropositive is still higher, but it's not up to the enclosed site. So this shows probably the enclosed environment enhances or increases the chance of becoming positive for COVID-19. And this will warrant the need for enforcement of the protection measures, especially in an enclosed environment. Okay. So we also looked at the age distributions among with relation to the seropositivity, where we found that individuals at the age of 20 to 30 years of age has the highest chance of becoming seropositive. And this is because these are the most active working age in the study site. This is followed by individuals who are at the age of 40 and 50. They're also part of the working age in the society with the least frequency seen in the individuals who are above 60 years of age. Okay. Also, we looked at the seropositivity based on the gender. Also shows that it is more likely to be seropositive if you are male in Nigeria. This is because probably the males has to go out, especially in the study site, has to go out to look for the occupation of the family. So they are more exposed to contact with the people, and probably they have the higher chance of having COVID-19 seropositive compared to female, especially in the study site. Okay. So we also look at the seropositivity based on the monthly incomes of our subjects, and the result shows that individuals at the low income class has the high chance of becoming seropositive compared to other income class. This is because probably these people, individuals at this class, even during the lockdown, they can't live at home because there is no food at home. They have to go out to look for the upkeeping for the family. So that's why the chance of them becoming infected is higher than others who can actually stay at home during the lockdown. We also checked at the employment status of the subjects, where the unskilled individuals tend to have high chance of becoming seropositive in our study site. This is because the study site, especially the markets and bus station site, these are individuals who must have to actually also be out to find means of survival. So the abiding by the COVID-19 protection strategies is very minimal among these individuals. They will have to come out every day to have the means of survival. This is followed by unemployed individuals. These individuals also dominated, especially the bus station and lorry stations, to look for livings. So, but it's much lower in among retirees. These are individuals that happens to stay at home, especially during the lockdown. Okay. We also looked at the educational levels of the subjects, where individuals that offer secondary school levels has the highest chance of getting sort of positive in our study site. This is because also there are also the workforce in the study site. But aside, most of the people at the lorry stations and bus stations, the market, they have the highest degree they have is the offer secondary school level, and then the chance of them getting seropositive is high because of the type of life they found themselves. Okay. Okay. Then we also looked at the antigen test results. For the antigen test, we conducted antigen tests in hospital site because we want to check the level of exposure at the hospital site. If we look at the antibody, we thought that it could be higher because the higher rate of vaccines and so on and so forth in the health institutions is higher. So we tend to check the antigen, which is more likely to show the active infections. And in the site we studied, this is Multidisciplinary Special Hospital, where we have the 8.9% of individuals who have the antigen, COVID-19 antigen. And then this is followed by IDH, this is Infectious Diseases Hospital, also in Kano. The lowest is an Kano teaching hospitals. These are individuals who presented to the hospitals with clinical conditions that are similar to that of COVID-19. Most of them are in the chest clinic, dot clinic, and so on and so forth. So there are individuals who are presenting to the hospitals for other conditions, not COVID-19. But they are found to be, quite a number of them are having COVID-19 infections. In total, there is 6.67% of these individuals who presented to the hospital for other infections related to COVID-19 that happen to be COVID-19 positive. Okay. Samples who happens to be...
Dr. Aliyu, your time is up, please. Can you have just a minute to run up for us? Thank you.
So this is the sequencing data from the ASF site. And then, So in conclusion, this is the first report of the large-scale population-based study for COVID-19 epidemiology in Nigeria. And we reported that COVID-19 in Nigeria, the epidemiology is varied from the gender, age, exposure site, level of education, and employment type. And the finding of this study shows that the average epidemiology of COVID-19 can be as high as 72%. This is quite much higher than what was reported using qPCR-based assays. And then a tenor of 75% level of immunity might warrant herd immunity, but this is something to actually watch because some literature say that the antibody against COVID-19 tends to disappear after a year or so. And the finding of this study Others suggested that there is need to have more robust system for timely data generation and dissemination in order to guide policies and public health strategies and disease management, especially for the future management of COVID-19 and other related infectious diseases. So it's our team in the different field. Okay, thank you very much.
Thank you. Thank you very much, Dr. Aliyu. Right, so we open up questions for him from the audience. Yes. Can I have the mic sent to him, please?
Thank you very much for your presentation. I noticed that you did sample over quite a long period, basically starting at a very early stage of the pandemic, December 2020, and the last samples were collected in May 2020, so just after the occurrence of the Omicron variant. Did you analyze your, one question would be, did you analyze your sample or in terms of when they were collected, how high the zero prevalence are? Because the reason I'm asking is that we had looked at one specific community in Africa and noticed that samples collected back in 21, that means before the Omicron wave, were around about what you reported, 50 to 60%. Yeah? And in the same community, samples were collected a year later, that means after occurrence of the Omicron variant, and was above 95% zero prevalence. I think it is very important, as you pointed out, to look for that. Thirdly, I would have a suggestion, a comment for rapid screening in populations for the presence of virus, because you depend on first looking at the antigen, at the antibodies. They may already have lost the virus, and until you have that, it's too late. What we did successfully just in our university to see how many people are positive at a specific site, because we want to see whether the university is a spreader, is that we collected throat washes. People are much more likely to provide throat washes, just 5 ml salt water, you gargle, and then you can pool a couple of samples and run them quickly through a reverse transcriptase with a one-step assay, and in a few hours you have an idea how high the zero prevalence is. If you're interested in that, we can talk about it later. But first of all, I would like to see whether you looked at the timing, what the frequency was.
Yeah, we actually did that. It's just that I didn't show the data. We analyzed the data before Omicron, and we also analyzed the data post-Omicron. And obviously, there wasn't a persistent pattern of like increase or decrease. The data looked somehow similar before Omicron and after Omicron. So that's why I decided to merge it. Even pre-Omicron, in some sites we had more than 67% X-ray positivity. At the same time, during post-Omicron, we have 70.1, 72. Somehow it's just similar, so we just took the average. But we actually have a separated time-based spatiotemporal sample analysis. Yeah. Thank you.
If I may add, because the point of you raised on providing information on herd immunity, there's also the possibility to test for blocking antibodies. Antibodies which block the binding of the spike protein to the ACE, recombinant ACE receptor. Apparently this surrogate test works quite well and gives you a good idea on how inhibitory the antibodies are. Referring to the study which I mentioned, where we looked in the communities, we saw that in August 21, before Omicron, only high level of antibodies were sufficient to block. After the Omicron wave, even much lower titers were able to block, possibly because of reinfection. So this way you can get more of an idea of herd immunity.
Okay, thank you.
Microphone, please.
Mike?
Mike.
Maybe, thank you for this interesting talk. Maybe a provocative question. How can this, according to the data you presented, we see that the majority of the population has been in contact with the SARS-CoV-2 variants. So how can this, and this is the case in many African countries now, how, according to you, according to the result, how can this inform vaccination strategy in Africa? There is one.
Okay. Based on the data so presented for Africa, the picture that is portrayed for Africa is that there is very, very low infection rate in Africa. And there is probably less need for vaccine, so to say, based on the data we have from the qPCR results. But obviously from what we have in this result and from the data we also produce, we show that probably that's not the true picture of the infection in Africa. There can be large number of subjects who are actually infected, who can also infect others, okay? But they are largely asymptomatic. So there is obviously need for us to vaccinate this group of individuals, okay? So actually the results shows that we need to do more in Africa as far as vaccine is concerned. Because the picture we used to see wasn't the actual true picture of the infection or the disease burden in Africa. Thank you.
Right, a question over there.
Sorry, excuse me.
Thank you so much for the presentation.
Okay.
2 questions. One, what do you think, what was the difference in terms of antibody and antigen detection in order to be able to see the actual infection? What was the rate of actual infection? And number 2, there is data that is coming out showing that actual infection protects more than the vaccine. So therefore, people who have been infected probably will be protected more than even taking the vaccine. So what is your opinion on that? Thank you so much.
Okay. Actually, this is what we said. I know the gold standard testing techniques for COVID-19 is RT-qPCR. But the capacity of these diagnosis techniques is less in Africa. Not only in Africa, but actually across the world. So if you are talking about the specificity, the qPCR is more specific, it's a gold standard. But affordability and then ease to perform, and even the specificity, because these are validated kits that performed more than 90% of the specificity and sensitivity. So they also gives you the true picture of whether you're positive for the virus, and it can also tell you the history. Probably you were previously exposed to the virus. You were asymptomatic, that's why you don't have any problem. But you were exposed to the virus. At that window, there's a window that you can infect others. Okay? So as far as large-scale testing is concerned, antibody is the best approach to go for COVID-19. But in terms of this can be supported by qPCR testing to confirm what we are seeing. And because of the possibility of you analyzing the antigen results to classify the subject into either they having previous infections, active infections, or the recent infections, this also helps you to classify the subjects who are qualified to go further for further analysis to confirm the result. With regard to the either if you are infected naturally, you are more protected or than the vaccines. Actually, if you are infected naturally, what is seen is usually the N protein antibodies in circulations. The most vaccines, the neutralizing antibody they produce is against receptor binding domain of the spike protein of the virus. That's the antibody produced against this domain, they are neutralizing antibodies. And these are the antibodies that are generated during vaccinations. It's more likely that when you are vaccinated, the antibody that are produced, they are more specific in virus neutralizations than in the natural infection. That's my...
Right, thank you very much. There's the last question online. So can we have online participants to ask the question, please?
Dr. Kamara, we've asked you to unmute to ask your question.
Sorry, I'm not seeking to ask any question for it. So my question was for the previous talk, so not that one, sorry, was a mistake.
Okay, thank you.
Did you hear the question? Right, so that's okay. Thank you very much, Dr. Ali. We are grateful. So our second speaker is Professor Koita, who is speaking on COVID-19 pandemic surveillance and network and monitoring, of population of SARS-CoV-2 in the network of diagnosis laboratories in Mali. Prof. Koita, your audience, thank you. You have 15 minutes, please.
Thank you. I would like to thank ICA partner for giving me the opportunity to come here and meet Pat Duffy. I knew many years ago. He worked with Don Krogstad, my mentor, who passed away 2 years ago. And they were working in two parts. Pat Duffy was on and to prepare the country for epidemics. Just a slide to show the toll of this COVID-19 compared to other pandemics beginning in 2000s. So MERS is about 850 deaths, and for COVID till February 28, 2023, almost 7 million of deaths. So we should be well prepared for the next epidemic or pandemic. I will not spend a lot of time describing this wording from WHO, considering SARS-CoV-2 as COVID-19, but we were hit by this virus from China, and Mali was in the epidemic in March 2020, and we got several deaths also like other countries. Here I would like to show how this is a severe pneumonia compared to human SARS-CoV infection and also the MERS. As you can see in this slide, sorry. Okay. I see now. Okay.
Can somebody help from the back, please?
Okay. I just want to see to show you the severe pneumonia caused by SARS-CoV-2. You see all the upper and the down. This part of the lungs are infected by the pathogens. If you have a coronavirus, human coronavirus, just the upper part of the lung is infected. So this may be the reason why virus is a recombine between this one and also between MERS-CoV, because you see the phenotype is quietly having a phenotype from human coronavirus. It was very complicated to protect older people and also people with comorbidities. And that's why this having a surveillance program is very important in our country. So the facility we have where we process the specimen from our study site is we was using the Sanger method, which is a capillary method, learned from our colleague from CDC. And we're using the CQ 8000 from Beckman Coulter, a gift from NIH. And also my colleague Xavier de Lamballerie from EHU, which also who gave us all those reagents to set up the COVID lab. And also we participate to Africa CoV ID, which was a consortium of countries participating to the surveillance of SARS-CoV-2 in Africa. We had other collaboration with some of my colleagues. These slides to show that when we analyze all the specimen using the RT-PCR, we found that CT, and the CT, when he's low, is a good indication about the burden of the virus. Here we find a CT around 10. And that prompted us to sequence this specific SARS-CoV-2 because we think it may be linked to some mutation, it may be a variant. That was our hypothesis because we could not sequence all the specimen. We just focused on this one because of a very short CT value. And then we followed the patient for about 3 weeks, which is normally in Mali is 7 days. But these patients had symptoms for about 3 weeks. The D0, when the onset of the disease was 16th of April. And we performed a clinical exam, an RT-PCR and a CCT. We repeat the same on April 24. And also till at the clearance time was May 16. 3 weeks, we follow up the patient. And then we try to sequence the spike gene of these pathogens and covering the entire domain of the spike genes. So when we align all our sequence that we deposit in the gene bank, we found it was very close to the B1525 from Nigeria and also from Italy. And he has 4 new mutations that we found in that part of the gene. And the sequence was deposited on the gene bank in April. And then we tried to compare, saying that the first wave that we knew in Mali, that may be due to the wild SARS-CoV-2 from China. And then this specimen was collecting during the third wave. And then we compare the mean CT between the 3 waves with our single specimen. And as you can see, the first wave, the mean CT was 30 compared to 10. And the third wave, so the mean CT was 25, seeing a big difference between. And that's why the conclusion of this study. Before that, and then we try also to see if this new mutation have changed the epitope, so to see if the vaccine can still efficacious on the SARS-CoV. We did an analysis was done by my colleague WANIDA group from EpiVax, and he showed that there is no difference in terms of change in epitope. Can be efficacious against this strain. That's a big binding between the receptor and our spike gene that we sequence. And to conclude this variant, and he led the third wave. Second, it was highly contagious because highly transmissible. And the way to manage a huge number of patients is to look at the CT value. If the CT value is less than 24, the physician should be very careful. He has to confine and follow the patient for maybe 1 or 2 weeks. That's why every time there is a positive, we should know the CT value. And that was a good indicator to make sure we can control older people. We have in Mali, because they were very careful, do not stop their activities because of COVID-19. So they put money. They asked us to build a P2 lab in 4 sites. So we have 4 companies exploiting gold in Mali. That was close to Guinea and is along the Sankara between Guinea and Mali. And the second site was the Siama site, is in the south, is at the border with Cote d'Ivoire and Burkina. Third site. This third site was Lulu site, which is at the border of Senegal, along the Falémé. So we conceived this lab for the third site. So all the 4 sites, we tried to put all the data was captured and sent to the Ministry of Health at the INRSP, which was the leading laboratory to manage all the diagnosis of COVID. So there was a partnership between the Minister of Higher Education and the Minister of Health. And that's why the response to epidemic should be across several departments. So in the university, we were able to bring our knowledge to make sure that the data are properly collected, analyzed, and sent to the database at the INRSP for the Ministry of Health. So then we participate to this consortium, Africa COVID ID, led by the Charity University in Berlin. Identify Omicron for Delta using the so the kits include the two markers, Delta and as you can see in Murila site, was no Omicron in September 2021. You remember that Omicron was described in November 2021. So in September, only Delta was circulating, no Omicron. And as you can see, the first case of Omicron was in December, normal. In Lulu, in June, you can see no Delta, but Omicron was there in June 21 before November. So 3 months before the appearance of Omicron in South Africa. And that's a point to show that the importance of a surveillance. And in September, nothing in Komana. And in Bamako also nothing. Donc we show that in June, Omicron was present in Mali, it was circulating. And the last slide to show, that's a scale of the continent, all Africa. As you can see, those Delta was present in, July, August 21. And Delta was present only in Mali in July 21. And that's critical because that will show you how we should be organized. Capture earlier any circulation from my colleague Aminata Maiga from Konji, and also NIH Bob Watts was very critical to put the lab in place. Tom Willems, your colleague, Dick Sakai, Cliff, and Sophia Siddiqui. I will not forget Don Crostat, my mentor, and also the city of Bordeaux for putting money to make sure we put all those data together. And
thank you very much,
thank
bro.
you.
We appreciate you. Thank you. Yeah. So can we have
Fred.
Detected, not in the
There was a case, one of our colleagues had CT of 11 and 18 and a month. Automatic.
patient who need oxygen. No. So that, but not severe. It was not severe.
You can have that.
Thank you so much for your presentation. I have a question regarding the project you are conducting on gold mining sites. Are you tracking or monitoring the surveillance of any other pathogen different from SARS-CoV-2? Are you surveying other pathogens, or it's just only focusing on SARS-CoV-2?
So I think your question is about if we are monitoring other SARS-CoV. Yes. Of the slides, it was not Delta, neither Omicron of another variant that we are tracking. So far, is not Omicron, is not Delta. And today we keep going, try to monitor, but it's not, all those strains actually in Bamako are not Delta, not Omicron. So at least our marker could not find actually which strain is circulating. That's why we see the third column sometimes is 1, 2. It was not Delta, it was not Omicron. Yes, the other SARS-CoV are circulating. And mostly SARS-CoV from base in the gold mine. We have data on that. We try to sequence them.
Right, thank you very much, Prof.
Thank you.
If you have further questions, you can grab him over lunch or over during coffee break, and then you can have a deliberation. So let's give it up for him. Thank you. So we have our next speaker, who is in the person of Dr. Diarra, who will talk about circulating emerging viruses in Mali. Right, so Doc, your audience. Thank you.
Ok, merci beaucoup. Moi, je vais faire mon intervention en français. Ok, permettez-moi de vous présenter les résultats de nos études de séroprévalence sur les virus Zika, chikungunya ou onyonyonko sur le territoire malien. Donc, d'abord, je fais une revue générale sur les 3 virus. Le virus Zika a été découvert dans la forêt de Zika, qui a même porté le nom de la maladie dans les années 1947. Et c'est un virus qui a été classé parmi les flavivirus, généralement transmis par les Aedes, mais aussi par la transfusion sanguine, la voie sexuelle et la voie materno-fœtale. Généralement, l'infection par ce virus est asymptomatique à 80%. Mais on peut observer chez les patients infectés une fièvre céphalée, des conjonctivites et des douleurs rétro-orbitales. Les quelques rares complications donnent l'intérêt à cette maladie, notamment les troubles neurologiques, le syndrome de Guillain-Barré et aussi l'enfant qui naît avec un faible poids de naissance et surtout une microcéphalie. Quant au virus chikungunya, le mot chikungunya désigne la maladie de l'homme courbé et le virus a été décrit aussi dans les années 1952 au Tanzanie C'est un virus qui est classé parmi les alphavirus et qui est transmis par les moustiques, la piqûre des moustiques Aedes. Contrairement au Zika, l'infection par le virus chikungunya est symptomatique à plus de 72%, où on peut observer effectivement des fièvres, des céphalées. Mais l'importance aussi de cette pathologie porte sur le fait que ça donne des douleurs articulaires persistantes, plus de 3 mois, pouvant même aller à 12 mois. Et surtout le syndrome de post-chic, où les malades sont souvent déprimés parce que ses mobilités sont limitées. Quant au virus Onyonyonk, il a été découvert aussi, et c'est un alphavirus qui est très proche du chikungunya sur le plan génétique structurel, avec une ressemblance de plus de 85%. Le problème, c'est surtout son diagnostic sérologique différentiel avec le chikungunya. Alors, pourquoi nous avons conduit cette étude? Parce que le Mali est un pays endémique paludisme, où le paludisme pose un problème de santé publique, compte tenu de ses taux de mortalité et de morbidité, et qui fait que la politique nationale de santé est principalement orientée vers cette maladie, le VIH et la tuberculose. D'autres maladies, surtout les pathologies virales, restent négligées à cause de ces grandes pathologies endémiques. Alors, il y a eu d'énormes progrès dans la lutte contre le paludisme qui ont contribué énormément à baisser l'incidence du paludisme au Mali. Mais nous avons constaté que malgré la baisse de l'incidence et de la morbidité, de la mortalité du paludisme, la fièvre reste toujours le motif de consultation le plus majeur au Mali. C'est pourquoi nous avons décidé d'investiguer les autres causes du paludisme, les autres causes de la fièvre non paludisme au Mali, notamment les causes liées à des pathologies virales émergentes. C'est ainsi que nous avons établi une collaboration avec l'unité des virus émergents à Marseille, dirigée par le professeur Xavier de Lambaléry, pour d'abord établir un programme de formation et de transfert de technologies sur ces virus émergents au Mali. Donc, l'objectif primaire, c'était de détecter la circulation de ces virus émergents et en fonction des résultats de séroprévalence, mettre sur place les sites sentinelles et de contrôle de ces pathologies virales. Alors, pour atteindre nos objectifs, nous avons conduit 2 études transversales sur tous les territoires maliens. D'abord, en 2016, nous avons conduit une étude sur 7 localités différentes du Mali, surtout dans les parties sud et au centre, des sites qui avaient des caractéristiques écoclimatiques différentes, mais qui représentaient les 2 zones climatiques différentes. C'était sur un total de 793 échantillons de sérum qu'on a pu récolter sur ces 7 sites. Et en 2021, nous avons conduit une seconde étude transversale pour couvrir la partie sec aride du Mali, qui, en 2016, on n'avait pas pu faire à cause de l'insécurité qui règne sur le pays. Alors, sur un total de 1640 échantillons de sérum, nous avons d'abord testé tous ces sérums par l'ELISA IgG anti-Zika, anti-Chic et anti-Onyonyonk. À partir des sérums qui se sont révélés positifs par les tests d'ELISA, nous les avons soumis à des tests de séroneutralisation des virus que j'ai nommés VNT. La participation des femmes était majoritaire dans notre étude. Globalement, dans la partie sud du pays, la prévalence du Zika pouvait atteindre jusqu'à 20% dans certaines localités et 15% aussi bien qu'au sud qu'au centre. Cette prévalence, heureusement, elle est faible dans la partie nord du pays où le climat est chaud et sec, 1%. Alors, en utilisant la modélisation mathématique, nous avons pu mettre en place 2 modes de transmission du virus Zika au Mali, une transmission endémique dans les zones savanniennes où 2,5% de la population serait infectée annuellement dans ces zones. et une transmission épidémique dans les zones semi-arides où théoriquement 18% de la population serait infectée dans les années 1995 suite à une épidémie qui est survenue dans les localités. Quant au contexte de chikungunya et de onyonyong, des virus génétiquement très proches, les tests de sérologie portent toujours des insuffisances pour une meilleure estimation de leur surprévalence. Et comme vous le savez, à cause de la ressemblance structurelle, il y a toujours des réactions croisées entre les deux virus qui induisent des anticorps et qui se croisent. Donc sur cette base, il y avait la théorie de l'asymétrie. Alors, donc, asymétrie, tout échantillon était considéré comme positif si le titre d'anticorps est supérieur à 40. Le titre d'anticorps de séroneutralisation. Et si ce titre est supérieur à 4 fois le titre de onyonyong, il était considéré comme positif. Le sérum est considéré comme onyonyong positif si le titre est supérieur à 40, mais aussi 2 fois supérieur au titre de chikungunya. Dans les autres cas, le sérum était classé comme équivocal parce qu'on ne parvenait pas à faire le départage entre chik positif et onyonyong positif. Et nous avons testé sur cette base de séroneutralisation les 1640 sérums du Mali. Nous voyons que ici la partie grise correspond à des sérums qui ont été classés comme équivocales, soit à peu près 10% des sérums que nous avons testés. Ce qui montre que l'ancienne classification porte des imperfections. Alors, nous avons pris, pour corriger ces imperfections, nous avons pris 62 sérums qui venaient de la Martinique et de la Guadeloupe et qui étaient uniquement chik positifs. Et que nous avons appliqué la méthode de classification ancienne. Nous avons trouvé que sur ces 2 îles, il y avait à peu près 16% des sérums qui étaient onyonyong positifs, ce qui n'est pas vrai parce que onyonyong n'a jamais été décrit dans ces 2 villes parce que le vecteur de transmission du virus onyonyong qui est l'anophèle n'a pas été décrit dans ces 2 villes. Ça conclut qu'effectivement, l'ancienne méthode d'asymétrie porte toujours des imperfections. C'est ainsi que nous avons établi une collaboration avec l'unité de modélisation mathématique de l'Institut Pasteur de Paris pour mettre en place un modèle pouvant mieux détecter l'infection à chikungunya et l'infection à onyonyong pour qu'en fait l'imperfection qui était dans les équivoques peuvent être améliorées. Alors, une fois mis sur place le modèle, nous l'avons testé par simulation avec 4 scénarios. Premier scénario, le chik est positif à 100%, donc 100% des sérums positifs à chik, 0% des sérums positifs à onyonyong. Notre nouvelle méthode mathématique détecte exactement 0% de onyonyong et 100% de chik, comparativement à la modèle classique où le chik revenait à peu près à 31%, contrairement à 100%, et 22% de onyonyong, contrairement à 0%. Alors nous avons fait 4 scénarios, et ces scénarios nous ont permis que notre modèle détecte mieux les chik et onyonyong que la méthode classique. Ainsi, sur la base de de ce modèle mathématique, nous avons effectivement trouvé que la prévalence du chik au Mali peut atteindre 20% dans certaines localités et que encore toujours au nord, dans les zones sèches, semi-arides, cette prévalence était faible, environ 3,7%. Globalement, la prévalence du chikungunya au Mali, en tenant compte des 1640 échantillons, était de 13,3%. Et que la prévalence de onyonyong au Mali était environ de 30%. Ça, c'est normal parce qu'il y a les vecteurs qui circulent sur tout le territoire malien. Pour conclure, nous avons, à partir de ces 2 études transversale conduite sur tous les territoires maliens, nous avons détecté la surprévalence, la circulation du virus Zika à 12%, chikungunya à 13% et oignonion à peu près 30%. Grâce à la modélisation mathématique, nous avons pu décrire des modes de transmission du virus Zika au Mali, endémique dans les zones savaniennes et épidémique dans les zones semi-arides. Les faibles taux de Zika, de chikungunya et oignonion ont été observés dans la partie nord chaude où le climat est chaud et sec. Je vous remercie.
Merci beaucoup. Merci. Right, so to the audience, questions for Dr. Diallo. Diarra, sorry. Questions from the audience? Yes, one hand.
Thank you, Prof. I have a question. I don't know if maybe you have the answer to this, but looking at, I think mortality from chikungunya is quite high, at least the official mortality rate. So I'm wondering if you have such a high seroprevalence. Is this something that translates?
Okay.
And then the second one is related. Zika, sometimes, I think usually people are worried about the second infection with Zika. Désolé. Okay, I'll start with Zika.
Okay.
I'll start with Zika. Zika, usually you worry about the second time you get Zika because it's supposed to be that time that you can get serious complications. So with such a high seroprevalence, is this something that is correlated with a spike in hospitalizations for Zika? And the first one was about chikungunya. Is it also correlated with deaths from febrile illness?
Ok. Merci beaucoup. Si vous regardez un peu l'introduction, je peux dire avec réserve que c'est la première étude où on a détecté d'abord la surprévalence des virus, les 3 au Mali. Mais il y avait d'autres études qui donnaient des surprévalences à partir des échantillons de sérum des banques de sang. Les cas cliniques, à mon avis, à ce que j'ai entendu jusqu'à aujourd'hui, il n'y a pas eu un cas clinique de Zika qui a été décrit, il n'y a pas eu un cas clinique de chikungunya, il n'y a pas eu un cas clinique de oignonion. Tout simplement parce qu'ils ont les mêmes symptomatologies que le paludisme et que le paludisme était considéré comme un problème majeur. La prise en charge de tous les cas de fièvre non diagnostiqués était soumis à un traitement antipaludique, un antalgique et un traitement antibactérien. Ce qui fait que c'est l'un de nos objectifs, c'est de découvrir qu'il y avait autre cause de la fièvre qui n'était pas que le paludisme et que peut-être ils vont donner un intérêt médical qu'on doit investiguer en 2 sur ces sites sentinelles en même temps. Merci. Oui. Ça, c'est une question culturelle et du comportement de la société. Au Mali, dans la plupart des cas, les femmes qui s'occupent des enfants, c'est les femmes qui s'occupent de la santé des enfants. Et chaque fois qu'il y a un problème de santé, quand on appelle la population, c'est les femmes qui se sentent toujours concernées. Donc, elles sortent toujours majoritairement. Et même dans les dons de sang, dans la banque de sang à Bamako, la participation volontaire des femmes, elle vaut 2 fois que celle des hommes. Donc moi je pense que c'est tout à fait culturel et comportemental.
Bonjour. Merci beaucoup pour cette présentation.
Oui.
Donc, moi, je voulais savoir s'il y avait des conditions environnementales ou des signaux qui favorisent le développement de ces maladies afin de pouvoir concevoir éventuellement des systèmes d'alerte précoce qui permettraient de capter des signaux avant qu'on ne puisse, par exemple, constater des périodes épidémiques. Qu'est-ce que vous pensez de ça?
Effectivement, la deuxième phase de cette collaboration avec l'unité de virus émergents au Mali, c'est la phase clinique, mais avec une équipe multidisciplinaire. Parce que dans la plupart des cas, c'est l'anophèle qui est plus étudié au Mali, à cause du paludisme. Et nous avons une équipe d'entomologie, un infectiologue, pour désormais investiguer, comme vous l'avez dit, presque les conditions environnementales, que ce soit entomologie. Et nous avons une équipe de géographie pour nous aider à faire la géoposition, tout ce qui est explication facteurs climatiques. Mais déjà avec la modélisation, vous avez vu que la transmission elle est endémique pour les Zika, épidémique dans les zones semi-arides du Mali. Et classiquement, on a 3 zones: savanienne, semi-aride et sec chaud. Donc il s'agit, avec une équipe multidisciplinaire, de mettre des sites sentinelles sur place, d'évaluer non seulement la prévalence, mais l'incidence de ces maladies, détecter les cas cliniques et voir leur co-infection avec le paludisme, quelle peut être la sévérité ou la cause de mortalité. Donc on en prévoit. On demande le soutien de WANIDA.
Dr. Diarra, and thank you, audience. At this juncture, I'll call on our next session chair, in the person of Dr. Podunu, who will take over from me. So the question online.
There's a question online.
Sorry. So, Dr. Diarra, there's a question online for you. So can we hear the person, please?
It's text form, so.
From France? Yeah. So thank you very much. The question is from Francine. She said, thank you very much for this nice presentation. You said that ONNV, onion virus, was not circulating in one study site because the vector Anopheles is not present. Do you not think that there may be secondary vectors acting there? Another question is, as I know, PRNT is a gold standard technique for serology. Is there no way of differentiating between a chick and onion infection? Additionally, why didn't you target also dengue virus? Thank you.
Je m'excuse de prendre les questions en retard, mais est-ce que vous pouvez les reprendre, s'il vous plaît? Ah, c'est good.
Okay, so come back to the first question. You said that onion virus was circulating in one study site because the vector is not present. Do you not think that there may be secondary vectors acting there? First question. So the second one is, I know that PRNT is a gold standard technique for serology. Is there no way of differentiating between a chick and onion infection? So finally, why didn't you target also dengue virus?
Ok, merci beaucoup pour les questions d'importance technique. D'abord, la première question. Comme je vous l'ai dit, en se basant sur les tests sérologiques classiques, comme vous l'avez dit, PNRT, Et en se basant sur les tests sérologiques IgG ou IgM, on ne pouvait pas toujours classifier les sérums à 100% chic positif ou 100% oignon positif. Il y avait toujours un nombre de sérums qui restait indéterminé. Alors ça, c'était basé sur la classification d'asymétrie. Alors donc, le site dont vous parlez, qui est la Martinique et la Guadeloupe, c'est là où on a pris des sérums où il y avait l'épidémie dans les années 1900, en 2014-2015. Il y a une épidémie de chikungunya sur ces sites. Ognonyonk, jusqu'au moment où on faisait les travaux en 2021, n'avait jamais été décrit là-bas parce qu'il n'y avait pas d'anophèle, c'est les îles françaises, il n'y avait pas d'anophèle qui avait été décrit. Donc, tous les positifs ognonyonk qu'on avait trouvés sur la méthodologie standard étaient des faux positifs. C'est pourquoi nous avons fait recours à la modélisation mathématique. Alors, la deuxième question, quand j'ai nommé notre test de stérilisation de virus VNT, Virus Neutralization Test, parce que c'est l'alternative qui a remplacé maintenant le PNLT. Et comme vous le savez, le PNLT, c'est un test classique ancien, mais qui utilise d'abord peu de sérum. Quand tu veux faire les tests, c'est maximum 6 sérums pour un essai. Et c'est très délicat parce que vous utilisez du gel à chaud, vous risquez quand même d'avoir des soucis avec les virus qui sont dedans. Notre laboratoire, le virus émergent, a mis cette nouvelle technique où, avec les plaques de 96, on parvient à tester plus de 88 échantillons. C'est rapide. Et deuxièmement, on n'a plus besoin de faire recours non seulement au colorant, le bleu de grenouille, on n'a plus besoin de faire recours au gel. La lecture se fait tout simplement en regardant les effets cytotoxiques des virus vivants sur un tapis cellulaire, comme vous le savez. Donc, c'est la nouvelle méthode qui a été récemment décrite, mise en place et validée avec mon collègue et on a publié le papier en 2020. Donc c'est la nouvelle, c'est l'alternative de PRNT qui est PNT. Et le titre de positivité, c'est que le titre soit supérieur à 40%. L'avantage de cette technique encore, c'est que ça nous a permis automatiquement, une fois que c'est positif au chic, c'est que les autres alphavirus, les autres flavivirus sont négatifs. On a testé avec des gens vaccinés contre la fièvre jaune, des gens qui avaient été infectés par la dengue, et on a appliqué cette méthodologie qui a vraiment fait la différence entre Zika positif, fièvre jaune positif, dengue positif. Donc c'est pourquoi nous l'avons utilisé comme nouvelle technique de référence.
Right, so thank you. And can we have our next session chair to take over from me in the person of Dr. Podono? Thank you.
My name is Jacques Podonou, and I'm your session chair for this session. We are running a little bit late, so I'm going to go ahead and get us started. Our first session talk is by Dr. Fermin Kra from the University of Alassane Ouattara, Côte d'Ivoire. Dr. Kra, you have the podium.
Okay. Good morning. My name is Fumekra. I am socio-anthropologist. Initially, I plan to speak in English, but how all the equipment is in place to translate, I go to speak in French on behalf of the francophonie. Donc, merci, je vais commencer ma communication en français. Alors, j'ai fait cette communication à la demande d'un groupe d'auteurs, puisque aucun d'entre eux n'a pu effectuer le déplacement. Donc, au lieu de faire la communication en visioconférence, nous avons fait le choix de faire une communication en présentielle à travers ma voix. La question qui va nous occuper ici est de savoir comment se préparer à prendre en compte les dimensions sociales des questions des épidémies ou des pandémiques. Pour répondre à cette question, nous vous proposons de partager avec vous notre expérience, l'expérience de notre réseau, le réseau anthropologie des épidémies émergentes. Voilà. Et comme vous le savez, les épidémies émergentes de ces 10 dernières années ont montré l'importance ou l'enjeu de la prise en compte des dimensions sociales des épidémies. Ce fut notamment le cas de la récente épidémie en Afrique de l'Ouest, l'épidémie de la maladie à virus Gasca, et comme les épidémies successives de la maladie à virus Ebola au Congo. Et plusieurs acteurs en sciences, plusieurs auteurs de santé publique sont aujourd'hui convaincus qu'on ne peut pas répondre efficacement à ces épidémies sans prendre en compte les questions sociales, culturelles et épidémiques. Et les analyses de nos expériences sociales ont montré qu'il faut évidemment adapter les mesures sanitaires au contexte culturel et politique. Et pour ce faire, nous avons deux réseaux en sciences sociales se sont mis ensemble pour renforcer les capacités des scientifiques sociaux, de sorte à pouvoir intervenir sur les questions sociales en contexte d'épidémie. Et nous avons le réseau anthropologie des épidémies émergentes, non, le RAE, qui a été créé en 2014 durant l'épidémie de la maladie à virus Ebola. Et il a pour objectif donc de partager les expériences entre les chercheurs. Et les premières activités de ce réseau ont commencé avec le programme PASS, le programme d'anthropologie comparée d'Ebola, qui a été financé par Expertise France dans 5 pays d'Afrique de l'Ouest. Ensuite, nous avons le réseau SONA Global qui est financé par l'Union européenne à travers son projet H de 2020 et qui est coordonné par l'Institut Pasteur à Paris. C'est un consortium de 18 institutions, et donc, dans le réseau anthropologique des épidémies et le CSRF. Et l'objectif de SONA Global est de créer une masse de scientifiques sociales, de sorte également à partager les expériences, les méthodes et les outils sociaux. Et donc, ces deux réseaux se sont donc mis ensemble pour mettre en place des modules de formation pour le renforcement de capacités. Donc, en 2017, en novembre 2017, les deux réseaux ont mis ensemble les dispositifs, ce qui a permis entre 2017 et 2018 d'élaborer des modules de formation pour le programme de riposte qui était initialement destiné à la formation de 250 personnels de la santé. Et sur 5 ans, ensuite, en 2019, avec les deux réseaux ont mis en place les modules de formation en anthropologie des épidémies émergentes. On l'a fait également en 2021, puis en 2022. Voilà, et ça nous a permis d'obtenir ces résultats. Donc, on a produit ces modules de formation, les 3, et celui-ci est sorti en 2022, vers la fin de l'année 2022, qui contient et vous pouvez les consulter sur le site des deux réseaux qu'on a créés. Donc, les modules que nous avons faits ont permis donc d'obtenir 13 chapitres, donc 13 modules de formation dont les contenus sont là. Ils abordent globalement les questions politiques, les questions culturelles et aussi la question du contexte d'émergence des épidémies dans le contexte sociales, les mesures sanitaires et les questions de finirait lors de ces épidémies. Donc, en conclusion, il faut dire que pendant ces années, nous avons donc mobilisé environ 27 collègues anthropologues dans 7 pays. Et en 2022, il y a eu, au-delà de Dakar, il y a eu une première formation au Burkina Faso et aussi plusieurs formations à l'endroit des professionnels de santé. Et le processus de la formation continue toujours. Et nous sommes toujours aussi en collaboration avec les autres pays de l'Europe. Maintenant, la question, c'est que c'est au niveau des pays pour que nous allons travailler ensemble pour que les autres pays puissent se l'approprier. Et pour l'année à venir, du 17 au 21 avril 2023, nous envisageons une formation de 25 chercheurs en sciences sociales à Madagascar. Voilà. Je vous remercie.
Merci, Dr. Kraft, pour une présentation, excellente présentation. One question, please. We'll take just one question because of time constraints.
Thank you very much. I think you're touching a very important aspect of fighting infectious and other diseases in this continent, to link social sciences together with infectious disease sciences. Do you have any concept in how to bring across such training modules, not only to social sciences, to anthropologists, but to people like those here in this room, beyond your presentation today, because I think this is a very good entrance to that. Thank you.
S'il vous plaît, la translation ne passe pas.
Translation?
Maybe unfortunately, I can't speak any French. I try maybe very slowly. My question was, basically, are these training modules also addressing people like here in the audience who are more working from the biology, from medical sciences, to link, end up linking social sciences with this kind of research which is described here? Did you understand?
Are you able to say the question in French?
Maybe somebody else can translate.
Yes.
Ok. La question est de savoir s'il existe des modules qui sont adaptés à des gens comme dans cette audience, c'est-à-dire des biologistes ou des chercheurs, etc., parce que ça serait très important et très intéressant. Donc, est-ce que vous faites des modules pas forcément pour les gens qui sont en sciences sociales déjà, mais également pour les biologistes et les chercheurs? Merci.
Merci.
Donc, si je comprends, la question, c'est la formation des biologistes. En gros, oui, je pense que dans le programme, il est envisagé des formations pour les biologistes, les professionnels de santé, mais aussi, on parle aussi des scientifiques sociaux, c'est-à-dire que des anthropologues et des sociologues, parce qu'on n'a pas toujours les méthodes adaptées en situation d'urgence. Donc, c'est pourquoi on fait aussi la formation pour ces personnes, pour que quand on est en situation d'urgence, c'est qu'on doit intervenir pour qu'on puisse adapter nos méthodes. Mais effectivement, il y a des formations pour les biologistes, les épidémiologistes, de sorte à ce qu'eux-mêmes ils puissent s'approprier les approches anthropologiques en situation d'urgence lorsque les anthropologues ne peuvent pas eux-mêmes être sur le terrain. J'ai oublié de préciser quelque chose, c'est que j'ai ma collègue Alice Desclous qui pourrait intervenir et qui est en ligne certainement.
Merci. Thank you very much, Dr. Kra. Okay, our next session is a roundtable on emerging health challenges and building surveillance systems within the One Health framework. I would like to invite Dr. Oliver Comey, Dr. Janet George Lorian, Dr. Benedicta Mensah, and online we have Professor Folaren from Redeemer University in Nigeria. So if you all want to come up. So welcome to this session. I'm going to just pass the mic to my co-presenters here to give a short introduction of themselves and their work.
Good afternoon. My name is Joseph Oliver Comey. I work with the Ghana Health Service, and I'm currently the acting director for the Ghana Infectious Disease Center. I'm an infectious disease specialist. I'm happy to be here.
Hi, my name is Benedicta Mensah. I'm a research fellow at the Noguchi Memorial Institute for Medical Research, and I am interested in looking at transmission dynamics of infectious diseases. Thank you.
Good afternoon. My name is Janet George Lorian from Sokoine University of Agriculture, Tanzania. I'm an assistant lecturer, but also a postdoctoral research fellow for SACI's African Center of Excellence for Infectious Diseases of Human and Animal. I was one of the first beneficiaries of AC2 in Tanzania and East Africa. So I'm so much privileged to be here and share my experience. I'm a One Health economist and systems analyst.
Thank you. Do we have Professor Folaren?
Yes, good afternoon, everyone. I'm greeting everyone there in Ghana from Nigeria. So I am Professor Onikepe Folaren. I am the Deputy Center Leader for African Center of Excellence for Genomics of Infectious Diseases, and we are based at Redeemer University. Nice to have you all.
Well, thank you, and thanks to our panelists. So the COVID-19 pandemic has reminded us of the inequalities of existing health systems across many African countries. Although significant progress has been made in building health surveillance systems, major systemic deficiencies still remain. No one can deny the close links between human health, animal health, and the state of the environment. Therefore, there's a need for greater collaboration between health research institutions across national boundaries to optimize the use of the available capacity to benefit all of us. So this symposium's first roundtable will have a presentation and a panel discussion on how do we deal with current and past pandemics and how to build better health, and most of all, to promote multidisciplinary to health issues. So with that, for me, a little bit about in building innovative transmittable disease, and can you also highlight some of the challenges that you're facing?
So thank you very much. I'm sure we were invited to this table because before COVID, most of the response in infectious diseases were done in silos. But then with the advent of COVID, we were privileged to have a center, an infectious disease center built to respond to future pandemics and future infectious diseases. So that's how come we come in. And before COVID, we'd never had a Ghana CDC. And I know we are moving towards trying to have a semblance of the Nigerian CDC. So we have a Ghana CDC also. So be that as may, the Ghana Infectious Disease Center is currently serving as the fulcrum around which we think that we should respond to future pandemics. As a center, we more or less focus mainly on the clinical management of the patients, but then we have a lab attached to it, and we believe that the lab is supposed to more or less serve the surveillance aspect of it. We are part of the Ghana Health Service Network, so we are open to everybody. Before COVID, most of our infectious diseases, we had to rely on the research institutions, either Noguchi, WACCBIP, to help us move things forward. And I think for where I sit, you highlighted, yes, the difficulties with research. For my institution, we can only be effective if we collaborate with the academic institutions. Going forward, we know that some of these infectious diseases are there. Currently, we are dealing with what we say is an outbreak of Lassa. I'm sure Lassa has been here for a long time, but we just haven't looked for it. Now we have an infectious disease center. We are managing the Lassa cases. We are collaborating with research institutions. It's not just about making diagnosis, but then we should use the modern, innovative ways, molecular techniques, sequencing. Recently we had Marburg, and we were told that the lineage of this Marburg virus was from Benin. It ended there. What else have we done? So I believe that it's important to make diagnosis, but it's important to understand or know the genomic makeups of these things. Once we know them, we can now look at how they interact with our environment. And we can look at how they interact with what's happening in one health. Then we can even model and predict some of our future epidemics and know that maybe, let's say, in the next 2 years, prepare to have maybe Lassa or an Ebola outbreak. Then we can pre-position ourselves to respond appropriately. So I believe that as the Ghana CDC, we need to position ourselves well and learn how to collaborate. Yes, we like infectious disease. One of the key things we are looking at is rheumatic fevers. How do we move that agenda forward? Is it just the ID people just talking about it? How do we collaborate with microbiologists? How do you collaborate with our friends who are experts in genomics to understand? And in future, even come up with vaccines, candidate vaccines, to manage some of these infectious diseases. So for me, I believe that the advent of COVID brought about a lot of, should I say, hope on the horizon for us in Africa. We should not limit ourselves just to the clinics. Let us collaborate wider, just not one country, but different countries. I'm just excited when I heard what the professor from Mali spoke about, using CT values to just predict the people you need to. And I think if we had such engagement, it will move us forward. You asked that I should add what are some of the difficulties. I believe sometimes for infectious diseases, the laboratory capacity. I always say, without the laboratory capacity, we are just making noise when we say we have infectious diseases. You need to diagnose them and know how they are behaving and how to manage them. For me, sequencing is the way to go. It's just not RT-PCR and we are there. Understand the disease, understand the pathogen, know the history of that pathogen, and then we can move forward.
Thank you, Dr. Comey. So if I get your summary, collaboration, multidisciplinary collaboration, and genomic capabilities. So this is, again, one theme that keeps repeating itself, collaboration amongst Francophone, Anglophone countries, sharing of knowledge, and how do we work as one team. So with that said, Dr. Mensah, you've done some outstanding work. Your team has done some outstanding work with the COVID-19. Can you tell us a little bit about what you've done and the way forward?
The ARIACOV project was a 2-year project funded by the French Development Agency to support IRD researchers and partners working in 4 West African countries, including Benin, Ghana, Guinea, and Senegal, and 2 Central African countries, including Cameroon and the Democratic Republic of Congo, in developing strategies, in promoting strategies to enable us respond to the COVID-19 pandemic. And one of the main objectives was to conduct field epidemiological studies and to also develop models of transmission. And in Ghana, we achieved this objective by the collaboration of IRD, Noguchi Memorial Institute for Medical Research, and WACCBIP. We conducted 3 repeated independent population-based surveys, and our results showed that the COVID-19 prevalence, seroprevalence, age-adjusted seroprevalence increased from about 14% to 40% within the 8 months period that we conducted this study. We also identified that people aged 40 years and above had a higher likelihood of being positive, being seropositive compared to the younger population. And we also identified during the 3rd survey that pupils or students had a 3 times likelihood of being infected as compared to unemployed people living at home. This was around the time where people have gone back to school and the transmission was higher in school children. Thank you.
Well, thank you. Your results are a bit similar to the Nigerian group that showed very high positivity for COVID-19. So what's interesting is I'm in the US, right? And the stories I'm hearing are that the incidence of COVID-19 in Africa is very low. So again, this is a scenario where we have to tell our own story, and the story shouldn't be coming from the West, telling the story. So I'm glad to see that there is capacity building happening in Africa, and hopefully you start leading the way forward. So thanks for that. Dr. L'Oreal, you've done some amazing work. in animals. How can we leverage, first of all, tell us about your work, and how can we leverage your work to better improve surveillance systems?
Thank you. Maybe also to give a context of how I arrived to that work. When I started my research, I wanted to have this beautiful One Health surveillance system, because Tanzania is among the very first countries to start implementation of One Health. We came up with a national One Health strategy. So we wanted to move to that direction of having One Health systems, because the role of surveillance, including also early disease detection, because the moment you have the disease, it becomes expensive to do the surveillance and control it. So it's better to have early warning system for that. But the roadblock that I encountered the very first time was that we have a stronger public health system, and our animal health surveillance system was not that much strong. So instead of now venturing into One Health surveillance system, I chose to go to animal health surveillance system. And then I started by looking on the status of animal surveillance system as it is in terms of operations, the data sources that we have, and I realized we have a lot of data sources that are not being used because maybe the structure of our surveillance system is mostly depending on the paper-based, but also we are depending on the livestock field officers and this official reporting line. So we are leaving a lot of data unused, which could have served as early warning system. So from there, I looked on the technologies that we have. In Tanzania, right now, we have among first countries also to test, to pilot MI, which is pioneered by FAO. And then also we had our digital, our innovation, the digital surveillance tool called Afya Data. So Afya Data is collecting data from the community and using syndromic approach, while the MI, which is FAO innovation, used case-based events. And then we also have laboratory information system. So thinking, is there a way we can have this integrated system? Because once we manage to make it interoperable, it's easier to see maybe in one dashboard what's going on in the laboratory, what's going on in the field as people they are doing their normal operations, and what's going on the daily reporting on the official line. That's when I integrated the three systems, and I could see that on the dashboard, although it was a prototype, but you could clearly see reflect all the three systems at once. So I managed to integrate to start with, to get we called API, the data, the formula that we are using to pull data from laboratory and data from the field, and then for MI. So to me, it was very, very exciting to see that at least now we can minimize the threshold. If the threshold to announce the outbreak is 100 cases per week, now we can reach that threshold in 1 day maybe. So we are able to detect the disease just on the onset, because you are having different level of alert. Maybe if we see several cases are appearing in different places, it's an alert. If you see cases the disease is endemic, but then the threshold value has increased, it's also an alert for us to follow up. But also we can have laboratory confirmation that this is a positive case and it's notifiable. So that way, with that kind of integration, to me, I was comfortable that now with the animal health surveillance as integrated as it is, can be an input to the One Health digital, to the One Health surveillance system. And then the next step now was we got a fund with our colleagues from Ending Pandemics to develop now the One Health platform, where now we are integrating the system from animal and human and all other early warning systems at country level, so we are able to respond as a One Health.
That's fantastic. Issues. Yeah, that's. It would be amazing if what you've done can also be replicated in other countries. What will you think we need to do to be able to scale what you've done? I mean, it's really amazing.
I'll go back to Dr. What he said, collaboration. We are trying to engage with the FAO. For instance, we have some countries that pledging to work with us, especially on integration of the countries that have implemented the MI and our innovation, the AFIA data, to integrate. So we are packaging this integration as a package, so that we start from the community. Those who are able to start from the community, they can do so, and then we have this official reporting line. But that can only be possible once we collaborate, because you can't, you may know share experience from other countries through collaboration. It may be less expensive compared if you want to hire a consultant to do that.
That's a very excellent point. Again, it comes back to collaboration and partnerships. Thank you. Professor Folarin, are you online?
Yes, I am.
Good afternoon from Accra.
Good afternoon from Nigeria.
So my question to you, your center has been at the forefront of sequencing for major transmissible diseases in West Africa. Can you please share with us your experience, and how do we optimize regional surveillance systems?
Thank you very much. I want to appreciate the organizers of this program and for inviting me, even though I'm supposed to be in Ghana physically with you, but due to some other engagements, I'm not able to be there. But in terms of our experiences concerning surveillance of infectious diseases, our experience actually started with Ebola outbreak in 2014. I'm sure everyone can still remember that. And we at SGID, we confirmed the first case of the disease. And then we also carried out genomic detection of other outbreaks that came up, such as monkeypox and yellow fever. And that actually brought us to start thinking that it's not going to take any special effort for any other pandemics to come into the country and also across the region. And so we knew that we have to start preparing for any other future outbreaks on a regular basis. So our presentation plan was therefore to build one, human capacity through series of genomics training, such as genomic foundation and advanced training that we do annually. And then we also have a periodic next generation sequencing training that we also train public health scientists, medical doctors, and also frontline health workers. And we also carry out bioinformatics analysis so that they can be available for genomic sequence data. So in all of this training, all the participants have hands-on training experience and real-time training in the field of genomics. The second thing that we have also done is to develop infrastructural capacity to give us an edge for high throughput genomic sequencing. And the center is presently equipped with series of equipment, such as the automation system for extraction and library preparation. And we also invested in the different next generation sequencing platform, such as the Illumina and as well as the Oxford Nanopore technology. So presently in the lab, we have the Illumina MySeq and NextSeq 2000 sequencing system, as well as NovaSeq 6000. So having these two in place, SGID was among the first generation designated testing center in the country for the detection of COVID-19. And we were also designated as the reference lab for detection of pathogens within the country and also across the region. So with respect to our experience on COVID-19, we actually sequenced the first case of the virus in the country within 48 hours of receipt of the samples. And this was possible because of the two factors that we have put in place, that is building human and infrastructural capacity. And so immediately we also provided the sequence data on GSAID for secondary data use. And as a follow-up to that, we equally sequenced over 7,000 samples, and we have also assembled over 5,000 genomes of the SARS-CoV-2, all of which are presently available on GSAID. We use the sequence data to define the circulating variants, emergence of new variants, as well as also to understand the transmission dynamics of the virus. And this is basically what we do for most of any outbreaks that we have. Then one thing that we also ensure that we do is we don't just keep the information to ourselves. We communicate the information to the NCDC, and also during COVID in particular, the Presidential Task Force on COVID-19. And this information have been used to inform policy towards the management and control of any of these outbreak. And aside of sequencing samples from Nigeria, I'm talking about COVID-19 now, we also sequence the virus from other African countries. We sequence samples from Cameroon, Sierra Leone, Angola, Rwanda, and many other countries. And we also did similar thing by ensuring that this sequence data were submitted to GSAID and the Public Health Institute in the different countries where the samples came from. So to answer your second part of the question, how do we optimize regional surveillance? And I think it's also going back to collaboration. So I would say that there's a need for cooperation among the countries and with the public health institutions such as Africa CDC, WHO, and they would help us to coordinate the surveillance process. This way, expertise will be identified and then knowledge will be shared across the region. And I would want to share our experience with COVID, what SGID has done during the COVID-19 pandemic. SGID, in collaboration with Africa CDC, WHO, we conducted training on next generation sequencing for the detection of COVID-19 for public health scientists and frontline health workers. And during this period, we trained scientists from Cameroon, Cote d'Ivoire, Seychelles, Rwanda. Overall, we covered about 32 African countries during this period. In addition to training, inviting scientists to come to SGID for training, we also went to the different countries to help them to establish their genomic laboratories. And we also trained them on site for COVID-19 sequencing. So this is what we need in Africa. Just like you rightly said, sir, African needs to tell their own story. And that's what our experience have been. We have been able to tell our own story, especially with COVID-19. Thank you.
Professor, that's very impressive work. I think it would be great. I mean, we have Dr. Komi, who is representing Ghana from the infectious disease standpoint. I think it would be great for all the countries that are represented here to perhaps during this 3-day session find ways to meet and to come up with a plan. You know, not just in words, but we should come up with an actionable plan for this kind of collaborations to happen. I mean, it's amazing work that's being done, and I'm really impressed, and I just want to make sure that this doesn't end here, that we all come here and have a nice meeting, and it all ends up here. So again, congratulations for such amazing work. Dr. Mensah. tell us a little bit about what Noguchi is also doing. I know you're doing some great work, and we want to highlight what you're doing and how you can also contribute to the sub-region pandemic surveillance program.
Similar to what Prof said, Noguchi is involved in research, surveillance, and also looking at promoting the One Health approach. So we have ongoing initiatives and programs, and one of them is the Emerging Infectious Disease Program, which focuses on identifying and response to new and re-emerging infectious diseases. So we conduct research on emerging infectious diseases such as COVID-19, Ebola, and Lassa fever, as he said. We provide capacity through next generation sequencing and also specialized diagnosis for Ghana Health Service. And we also support the Ministry of Health. And we actually house the national, we are the reference center for polio nationally, and then regionally we are the center for influenza. I haven't switched that up. And then we are also, we also have the One Health surveillance approach where we focus on a comprehensive surveillance system, as she also described, where we integrate human, animal, and then also environmental data in detecting and also response to outbreaks in a timely manner. We are also interested in looking at antimicrobial resistance as well. We want to create awareness in the public. The aim is to create awareness in the public about the use of antibiotics and also develop strategies to combat antimalarial resistance, and also develop new antimicrobial agents. And then we are, finally, we are also interested in vaccine development. We've been involved with vaccines towards Ebola, and we also have a specialized facility where we are involved in phase 1 and phase 2 trials on vaccines like COVID-19 and malaria.
And by Noguchi. Now my final question goes.
What this organization stands for, WANIDA stands for. I will not be speaking for Ghana, because then if I speak only for Ghana, I think that my scope will be limited. But being here and seeing the capacity in other countries, I will go back to my question of saying, let us collaborate. We will sit down, come up with a plan. We cannot focus everything in one country. So let's say Senegal is going to handle for us maybe vaccines, Ghana handles for us diagnostics, Nigeria handles for us NGS sequencing. Then we have the rich experience from Mali telling us clinical things. I'm sure if you ask me, you have the money, I will sit down with my colleagues. We'll come up with a plan that will maximize what we want to do to benefit everybody rather than just one small country.
That's very well said. So in conclusion, I think what I get from this panel is this very exciting discussion. And again, want to thank the World Bank folks, the IRD, and the French Development Bank for helping put this symposium together. Thank you. I guess lunch is next. Thanks.
Another. Another round of applause for our panelists. Okay, it's time for lunch, but before we go, a few housekeeping items. You may have noticed in the program that we have a networking lunch with HCA. Going on at this very moment, we have a workshop by the Human Cell Atlas group. And the idea is that we will share the lunches so that there's collaboration between, there's conversation between the groups that are sharing this time and space at WACCBIP. And then maybe some ideas will come out of some of those conversations. Some of our faculty and students are involved in that, and some are involved in this. But we couldn't find any other way to sort of merge the two events. So hopefully this lunch will be that. And lunch will be served outside in the car park when you exit just to the right. There's also posters which are on the first floor in the main biochemistry building. You've moved them? So for the Americans, second floor. For the British and Ghanaians, first floor. So I think that's it. And we'll meet back here at, what time? 2:30. Thank you. Bon appétit.
then when the gametes come out of the red cells in the midgut, that's when the antibodies can attack them when they come out. It's really fascinating. That started in the 1970s, the concept of the transmission. Yeah, shall we walk over there? You can leave things to be said, Oh, thank you. Back.
Est-ce que on a la télécommande de ça, juste les temps 30 secondes?
Should I put it up here?
Yes. Okay. Perfect. Thank you. Thank you very much. Okay. Okay. Thank you. Okay, here we are. First of all, could you introduce yourself?
My name is Joseph Olilakome. I'm the acting director for the Ghana Infectious Disease Center. I'm an infectious disease physician by training. I love infectious diseases.
Okay. Could you tell us how was table going on? Was it good? Was it interesting? What did you talk about?
Okay, so I took part in the plenary on how we can maximize surveillance to look at infectious diseases. I must say that I was impressed with what I heard from my colleague from East Africa and also from Nigeria, and also the work that is being done in Noguchi or WACCBIP. I believe that the key word here for me is we need to collaborate. If you remain as a one center, you are not able to achieve a lot. But then when you collaborate with other centers from different countries, what you think is a difficulty for you, somebody might be doing it very easy. And what that person thinks is a difficulty, you may. So we can harness our efforts together. And I'm sure we can have a solution to an African solution to the infectious diseases that really worry us more than any other place.
Okay. I'm just thinking if I have another question. Why is it important for you to be part of this WANIDA Symposium?
Okay, I must say that maybe I stumbled into this symposium by accident. I happened to meet Olivia, and then she said, you need to be here. I read about WANIDA and what you are doing, and I must say that having centers of excellence or aces of IT is key. For my center, we are just 2 years old, and I'm sure we will look forward to growing and to be considered as part of the ACE ID center. So for me, coming here, maybe we will say it's an accident, but I'm happy to be here and seeing what others are doing elsewhere. It gives me great motivation that if we work hard, we shall also get there.
Okay, that's okay for me. Thank you very much.